And while we're waiting for other people to maybe show up, are there any questions, logistical questions, or anything I can answer? No? We're all good for, yes?
Can we go through the period histograms at some point?
Yes.
How to build one?
Yes, we'll definitely go through that.
Thank you.
Yeah. The point of the review session and how to use it is that we're gonna cover the main topics, but since there are a lot of topics, we might not be able to go in detail about every one of them. We'll definitely touch on them. And yeah, the best way to study, the way I studied for SBE exams, are to watch the lectures if you have time. Or like watch the ones that seem confusing and you don't completely get. Okay, so I guess we'll just go ahead and get started. So I'm going to cover the psychophysics and the receptive field, and then Ananya will cover the rest of the Dr. Wong's section. Feel free to stop me at any point if you yes.
Just can you speak up a little bit?
Oh, sorry. Feel free to stop me at any point if you have any questions. I'll try to speak loudly. If you don't hear me, just raise your hand or tell me at some point. Okay, so why do we want to use psychophysics? The reason is that what you see in a stimulus, like the physical property of the stimulus, like its frequency, does not necessarily translate to how you perceive it. So you might think something is loud. The loudness doesn't necessarily mean its intensity. So there's a gap between our human perception and the physical property of the object. So we use psychophysics, which tries to quantitatively relate the behavior of the person to the physical properties of the stimulus. It's important for you guys to know these definitions by heart. Okay. Now, what is psychometric function? Psychometric function is the data for psychophysics. Hence, it relates quantitatively the subject's behavior to the physical stimulus. And it's a quantitative measure, remember that, and it measures what? It measures behavior. So the x-axis in a psychometric function would be the stimulus physical property, and the y-axis will be your behavior. Like, oh yes, I saw it. Oh, I didn't see it. So. And it's going to look sigmoidal most of the time, as you saw in your homeworks. Any questions about psychometrics, functions, or anything? All right, simple. Okay, so we have two different types of psychometric functions. One is the detection. So you're either detecting whether a stimulus exists. So like, oh yes, I saw that black box. Or you're detecting a difference, like, oh yes, I saw a difference between these two black boxes. I know they're different. Or, oh no, I don't know they're different. So that's the first type. That's the detection of a stimulus or a detection of a difference. Now the next type is the two forced choices, which means like, I give you two boxes, and they look identical color-wise. But I ask you which one is the lighter one, and you have to choose between them. Although you think that they're different, so you see how that's different from the first type? The two AFC, the alternative forced choices, makes you choose between them, even if you think that they're not different. So it makes you choose which one is the darker one, which one is the lighter one. Hence, when their difference is like 0.00001, you'll obviously not see that difference. So when you have to choose between them, you're most likely going to be 50% right. So you're going to choose the right one 50% of the time because you're just guessing. Okay, so let's go through each of them in very detail. Okay, so the detection one asks you, do you see a black box on the screen? And you can say, no, I don't see it. Or you can say, yes, I do. So it's going to go from 0 to 100%. When it's very light and it's barely detectable, you might just say, no, I don't see a box on the board. So it'll start at 0. And then when it's very dark and you definitely see it, then you'll be like, yes, I do see it. So that's 100% detection. And so now we can think about thresholds. What's your threshold of detecting? We'll go to the 50% of the time. And the stimulus physical property, this particular value over here, that's your threshold. And it's called the absolute threshold in the case of detection. Okay. Now for the two alternative force choice, again, you will be right 50% of the time at first when there's barely a difference between the two boxes that are presented to you, because you really can't detect any difference between them, so you're just guessing. That's still going to go to 100% when the difference is obvious, like in this case. You're going to most likely pick the darker one because you see the difference. So that's going between 50 to 100%. And the threshold, which is now going to be relative, all of that is on top of that, by the way, but I just like using board. OK, so the threshold that's now going to be relative is going to be at 75%. Yes?
Do the units on the x-axis always have to be in block units, or?
No, you can have it in any units you want. Any other questions?
Why is it at 75%?
Because you have to go halfway between your starting point and the 100%. And your starting point for two alternative force choice is 50%. Okay. Any other questions regarding this? Because this yes.
That like minimum 50% represents like random guessing, right?
Yes. 50% is because of random guessing. Yes.
So if the question is, are two things different or the same? It's that category, right?
Yes.
Okay.
Yes. So detection of a difference is when I ask, are these two boxes different? And you can say no, they're not different. Hence, you start with a 0. But the two alternative force choice, I tell you, pick the lighter one. Although they might not look different to you, you'll be wrong 50% of the time. So you're guessing, yes?
Okay. I fully agree with you. That makes sense.
Yes.
But why is in lectures he says that is an example of a...
Discrimination test.
Yes.
Yes. So what he means is the discrimination, okay, the discrimination test has two types, the difference types, and it has the two alternative force choice. So if you go through his lectures might be a little bit confusing because he seems to group this one with the discrimination section, but that's not the case. He's talking about two alternative force choice here, and for detecting whether something is different, and if you have the option of saying no, they're not different, then that becomes a detection test. Yes?
So if you're doing a detection experiment, is it like all of the boxes are always different, but the observer doesn't know that?
So you technically start at 0, but you may think of it as starting at 0.00001, so that the test subject will really not be able to tell the difference between the two, because it's...
The boxes are always different. Like there's not going to be two boxes that are exactly the same during the test.
Yeah,
Okay.
you can think of it that way, yes. Any other questions? These are great questions. Okay, just keep in mind the detection of difference is different than pick the darker box. Okay. Now we can talk about increasing sensitivity or lowering your criterion. So increasing in sensitivity, it occurs when, like, for example, we're doing an experiment on your hand and you place a pen. Like I barely touch a pen to your hand, you might not feel that. But if I barely touch a needle to your hand, you'll definitely feel it. So you're more sensitive to certain objects versus other ones. Or like if you're heightened or whatever, if you just came from running, you might be less sensitive. So changes in sensitivity occurs, And the result is that your threshold either increases or decreases based on how your sensitivity changed. So if you become more sensitive to a stimulus, you're going to shift this curve to the left because you'll be detecting it more often. So that's increased sensitivity. It brings down your threshold because you're more sensitive. Now, what is criterion? If you remember the beta that we talked about with the two curves that look like this, and you had a beta and your beta moved, your beta is your criterion. Basically, it means how picky you are. If you're very picky and particular about what a stimulus is, you're going to detect it less often because you're going to be like, that didn't match all of my expectations, so that's not a stimulus. But if you're very picky, if you're not very picky, you're going to detect it more often. So that's basically, you can think of it as being picky. So shifting that way, meaning if you are detecting it more often, that means you're less picky. That means you're lower criterion. You have lower criterion. Does that make sense? Yes. This way. Same way as increased sensitivity. Oh, okay. Beta would also shift this way because your hit area becomes larger again, and you call more things the stimulus even if they are the noise. Yes?
This curve doesn't have anything to do with the false positive rate, though, does it? Like this doesn't tell you that.
It doesn't tell you that from here, no. But if you are lowering your criterion, you are increasing your false rate, which can actually be asked about during an ROC curve drawing. We're getting right there if there are no other questions about it. Yes?
Would it be correct to say that criterion is a function of like what level of neuronal firing causes someone to say that there's a response versus sensitivity just being like whether or not a given stimulus evokes a particular level of neuronal firing?
Yes, that will be perfectly fine definition. And for those of you who didn't hear him, he said that the criterion is the level of firing that you call a stimulus. So do you call 10 spikes per second a stimulus or like 20 spikes per second? Okay. All right. So now we can talk about these curves, which then give you the ROC curve. So this axis is your observer criterion varying. So it's like you can think of it as your neuron firing rate. For the noise, you can think of noise as basically how much your neurons are firing in the resting stage, because they have to fire to keep themselves alive anyway. So you can think of the noise as how much they're firing just in the resting stage when there is no stimulus present. And then there is the stimulus. Which is going to be shifted to the right because you're going to be increasing. We're going to think in this case that we're increasing our firing rate in the presence of a stimulus. So, so now there comes the concepts, as one of your classmates pointed out. There comes the concepts of hit, miss, false alarm, and correct reject. So for the hit and miss, we're going to think only about the stimulus curve. So if this is my beta, my hit is going to be under the stimulus curve. And my miss is going to be under the stimulus curve. And then alarm is like when you say there is a stimulus present, but there's actually nothing but noise. So your false alarm is going to be over here. And then correct reject is when you say there is nothing present and there's actually nothing present, so that's correct reject. highly recommend you guys go and look over your homeworks, because that gave you an example of how you can potentially be tested on a you know, on an ROC curve type of question. So the D prime measure. Sorry, is there a question? Oh, D prime. Yes. The D prime is the distance between these two curves. So we are gonna assume, for the sake of like this class, that these curves have the same variance. So the standard you know, error is the same in both curves. So this is the same for both of them. Now, big D is the distance between the two means. So the D prime is D over the standard deviation, right? So it measures how far these curves are from each other. The further they are, the larger the D prime. So farther, larger the D prime. And you'll see that in the next slide. As you can see over there, that's D prime of 1. There's a lot of overlap between the two curves. And the D prime of 3, not much overlap because they get further apart from each other. Which makes sense because D prime is D over some constant. Yes?
On some of the past exams, there are questions where like they'll give us the sigma and then they'll tell us like basically where the two curves or their means are. And then they'll ask what's the probability of false alarm, what's the probability of correct response.
Yeah.
Do we like just memorize some of the values for different D prime or like?
No.
Okay.
No. So, yeah, don't ever memorize. For ROC curves, you always have to calculate it on your own. So let's say they tell you that they are uniform distributions. They're not going to give you very difficult things that you can't calculate without areas or anything. So like he's either going to give you a uniform or something simple. So let's say it's uniform. And then he gives you the D prime, which is the distance between the two. Oh, I'm sorry. He gives you the big D, which is the distance between the two. And then he gives you the variance. Like, let's say 10, right? Then you can actually calculate the area of like hit or whatever based on your beta. So this is your observer criterion. So to calculate like, the hit, let's say if you want to calculate the hit value in this case, you're just going to find the area under the curve here, which can be done either using a PDF to CDF type of function, because you have the PDF of a uniform is 1. So you calculate the area under the curve of this guy. So like, for example, in this case, it might be beta, from beta to like infinity, but like it's not infinity because this curve probably stops somewhere, right? So let's say it stops at 50. I don't know, I'm just making up these values of like the PDF. And let's say if this started from 0 to 50, then it would be 1 over 50 dt or d, whatever. So like, you get the idea. Do you get the idea? So it would be like, does this make sense? Yeah. I'm trying. Come talk to me after if it doesn't. But yes.
Basically, other than beta and the values that like 0 to 50, it's not super necessary to have D or standard deviation in this specific problem.
Oh, yeah. I mean, he gives you D for you to figure out where this curve is going to be, right? Because if D is larger, this noise curve is going to be this way. So then you can find the false alarm based on how far these curves are from each other. If it actually helps you guys, I can make up an example and post it to Blackboard. Okay, I'll make up an example, post it to Blackboard. Okay. Now coming back to this, coming back to this, we can actually draw ROC curves based on moving your beta. So as you can see, if we move our beta from beta 1 to beta 2 on the curve above, like the two curves above, if we move from beta 1 to beta 2, you can see that the hit region is becoming smaller, right? Because the hit region is the area under the stimulus curve to the right of the beta. So my hit region at beta 2 is smaller than my hit region under beta 1. Hence, my point comes down in the ROC curve, right? Because we have lower values of hits and we have lower values of false alarm. Now, ROC curves are drawn using, can I do this? Ah, yes. Okay, so ROC curves are drawn using like a tens or 20s of like different betas and calculating for each of them what's the value of hit and what's the value of false alarm. So you have to calculate the area under the stimulus curve and the area under the noise curve to the right of the beta. And that's how you draw ROC curves. Now this line represents when you don't actually have any difference between your stimulus curve and your noise curve, meaning they're exactly right on top of each other. So let's actually like go through that one, because if you understand that one, then you can understand all of them. So if your D prime is 0, that means that your curves are identical. This is both my noise curve and it's my stimulus curve. So for whatever value of beta I choose, this area is going to be both my hit area and it's going to be my false alarm area, right? So my hit and false alarm values are always going to be identical if my D prime is 0. Hence, I get this straight line, right? Okay, any questions? ROC curves will be, yes?
So I feel like in lecture, it was described as, so you described it as the noise between the fire rate and the fire rate, but I feel like it was described as the noise in the stimulus, or in like the background of wherever the stimulus is presented. Are we supposed to think of that as like equivalent, or?
So they're not equivalent because firing rate, again, is your perception. You can think of it as the perception, and then the stimulus is your physical property of the object. But there is a correlation between the two based on the psychometric function. So you can draw these two curves based on the stimulus, or you can draw these two curves based on firing rate. Good questions. Any other ones? Okay, I'm sorry. All right, so moving on to receptive fields. Receptive fields is the physical area on your skin or in hearing. It could be anywhere, but it's where the stimulus actually modifies the firing rate of your neurons. So in this example that he provided, if you touch to the center of this receptive field of this particular neuron, if you're on the center, it increases its firing rate. If you touch around it, it'll decrease it. And if you're completely outside the detection area of this particular neuron, you don't change its firing rate. Basically, it just changes the firing rate of your neuron, of that particular neuron. So he then talked about the auditory neuron. Yes?
There was a question in the past exam that said something like, does the receptive field of a neuron depend on how we measure it? And what's the answer?
Yes. Yeah, it does, because it depends what you call it depends how you measure the firing rate. Yes.
Doesn't it have to do with how you choose your criterion for whether like you have a stimulus? Is that what that question is asking?
Yeah, you can think of it that way. You can also think about it as like, or maybe not. I don't know. Any TAs want to chime in here?
So question from past exam says something like, does the receptive field of a neuron depends on how we measure it? Something like that. I can give you exact.
How we measure?
Wait, can you
Does
give
the
us
size of a receptive field depend on how you measure it? Why?
what year is that? Like, which one? Or does it give a
217.
Okay. I'll look at the question right
So, yep, we'll get back to you on that. Okay, so the receptive field of an auditory neuron. Okay, so the receptive field of an auditory neuron, here you have a tuning curve, which I had drawn, but I regressed. So how do we actually draw tuning curves? You actually use the psychometric function. So you run a bunch of experiments, and you use the psychometric functions to draw the tuning curve. Each of these points on this tuning curve is actually a threshold that came from a particular experiment. Each of these points are thresholds that came from a particular experiment that you ran using psychometric functions. So each of these points are actually 50%. It's like the value at which you detect it 50% of the time. So like if this is if this is like your frequency and this is your decibel, at this particular frequency and decibel, yes?
Wouldn't that just be true of an audiogram? Because for a receptive field, you're not looking at an observer's response. You're just looking at whether the neuron is...
Yes. Yes. Sorry. This is for the audiogram. This is how you draw the tuning curve of an audiogram using an experiment, using a psychophysics experiment. Yes, if you have the ability to like put stuff in someone's ear and like measure the firing rate, you don't use the person's response. But if you can't do that, which is most likely the case, you use their response as, oh, I heard it, oh, I didn't hear it, as a way of like measuring their receptive field. Yes?
So with this one, it would be the same frequency, you'd just be changing the intensity, and then you take the 75% threshold to hit it over there.
Exactly. So you fix the frequency in this experiment at like 10 hertz. And the thing that you vary here is the decibel. And once whatever threshold value you got here is the value over here. See, this axis becomes this axis over here. And then you change the frequency, you change like the experiment, and you measure it again to find these frequencies and to find these thresholds. And then you draw your tuning curve. And your receptive field now is going to be anything higher than that. Remember, because we're going higher than that, that's detection. So like this area is going to be the receptive field that you measure for your auditory nerve. Then yes, you can do that, but that might not be the case all the time. Oh, and then the characteristic frequency is when you're most sensitive to the noise. So over here you have the characteristic frequency. Okay, yes?
If we ever have to draw a receptive field, can we just shade in the entire area encompassed by the curve, or do we have to actually draw it?
No, that triangle is just a schematic because I couldn't shade it. Yeah, it's everything above that curve. It's not a triangle. That's just PowerPoint. Yes?
So for the characteristic frequency,
Yeah?
technically it's like a different noise, like, because you're changing the frequency value. So like, based on the prior example, say like 1 hertz,
Yeah?
like detecting a frequency of a noise at a frequency of 1 hertz.
Yeah?
If you change it to 2 hertz for the characteristic frequency, it's something.
No, characteristic frequency is when you're most sensitive. It doesn't have to do with the thresholds. So you use the thresholds and you drew this tuning curve. And now here at the bottom of this curve is going to be your characteristic frequency. Each neuron is going to have only one characteristic frequency, which is basically when they can sense the noise the most.
Okay, so it's like that neuron is best at this noise.
Exactly. Yes.
So with that in mind, if we're asked on the exam, for example, to like describe an experiment where we measure the receptive field of a single neuron,
Yeah.
we wouldn't do the audiogram approach. We'd assume that we can measure single neuron firing.
I would use the audiogram approach Because, well, you can say, okay, if you can actually measure the fire, you can ask in the exam.
Because otherwise you wouldn't be able to measure the receptive field of a single neuron and generate the single neuron's tuning curve, which is what that figure shows. That's
true. Yes, because that would just be the personal, yeah, true. You just use that. Yes, if we ask questions regarding this in the exam, you will be given all the details you need if you don't ask us during the exam. Okay, so remember this, when you lowered your criterion, you shifted the psychometric function. So if you are shifting the psychometric function, you're shifting the threshold with it. So if your threshold is becoming lower, that means the tuning curve is shifting down. Got it? So if you're decreasing your expectations, you're becoming less picky, your threshold is becoming lower, your tuning curve shifts down. Your receptive field gets larger. That's how we define it. And I think this has to do with that question. Receptive field depends on how we define it. As you can see here, if we are decreasing our criterion rate, the receptive field is getting larger. So it depends on how you define it.
Wait, can I add on to that really quick?
Yeah.
So in the Wang Lecture 2 handout, slide number 7, I think that's your answer. So.
That was very Wait, can you say it again? I'll repeat.
Yeah, so it's Wang Lecture 2 handout, the 7th slide.
Okay, Wang Lecture 2, slide 7. I'm just saying it for the microphone. Lecture 7, slide 7, Lecture 2 is the response about the receptive field, whether it depends on how you define it.
And it's like what you just said, basically.
Oh, it's what I said. Okay. Now resolution. So this is important to know because it also relates to what Connor is talking about, because Connor talks about how different, bipolar cells might synapse with multiple or few rods and cones. Rods and cones. So this relates to his lecture, which we'll touch upon later. But basically, it means that the density and the size of receptive field determines the resolution of the image. So if you have higher density of receptors, you get higher resolution. And if you have smaller receptive field for each of them, you get higher resolution. Remember, Wang talked about touching like someone's back versus someone's face. The face, your neurons have smaller receptive fields, so you have higher resolution or sensitivity of detecting whether 2 points are different. But someone's back, you don't have, your receptive field of each neuron is larger, so you have less resolution or you have less ability to detect differences between 2 points. Any questions? All right, then we're done with my part. Nani? Who asked? Someone asked that. Okay.
Can I get the mic?
She gets sorry.
Sorry?
Sorry, what did you say?
The mic.
Yeah.
Where do I U-turn? Wait, so this just goes
On your shirt or something.
okay,
And then the other one goes in your pocket.
like that. Like that. There we go. Perfect.
Okay guys, so
So I'll cover the rest of Dr. Wang's material, which includes analysis of action potentials, so the different ways to process them to make different graphs, as well as sort of the auditory system biology that you need to know of, like how sort of at different levels of processing you have sort of the auditory nerves up to the cortex. Any questions? Yes?
Is the Snow Day lecture posted on Blackboard, or is it just completely not posted?
Oh yeah, so the Snow Day lecture on Feb 20th of Dr. Wang, that is not being covered on this exam. So you do not have to go over that. Yeah. Any other questions? Okay. Yeah.
One of the lectures like tells a load on Blackboard. It might just be me from.
Okay. Come talk to us about that afterwards and we can fix it for you. Any other questions? Okay. Let's get started then. So, okay. So first of all, let's just talk about action potentials. So action potentials are spikes, and a sequence of action potentials is called a spike train. And so the reason we call them spikes is because action potentials are all or none. So as you can see there, there's all or none coding of action potentials. Either they happen or they don't happen. It's not that they happen halfway or to different intensities. Action potentials are all or none, so we can just represent them as spikes. And so each spike there actually just represents an action potential. And this becomes important with the processing, with how action potentials are represented in graphs. So how to interpret a dot raster. So dot rasters can be kind of confusing because typically what you see in the slides looks something like that. What this means here is basically each dot is basically just an action potential. So going back to this slide, how to interpret a dot raster is basically just that. Imagine there is one single neuron that you're looking at and you're presenting some kind of stimulus. These slides will be posted on Blackboard, by the way. There is some kind of stimulus that you're giving at time 0. You're just looking at, okay, in trial 1, after the stimulus was presented at time 0, how does the neuron react in terms of action potential? So at what time points is it sort of firing? And so you're looking at that data across multiple trials for the same neuron. And so that's basically how a dot raster works. So what we can take away from a dot raster is mean firing rate. And so if you were to just look across one trial, what it basically tells you is, okay, over a time period of, let's say, 500 milliseconds, this is the number of times that this neuron has fired. And so you can calculate mean firing rate by just doing total number of spikes over time. So total time over here, so 500 milliseconds. If you were to do this experiment across multiple trials, then you would have to basically count all of the spikes in the dot raster, divide that by the number of trials. So basically you're averaging, you're trying to find the average number of spikes per trial, and then dividing that by the total amount of time to get mean firing rate. Any questions? Yes?
Is presentation number the same thing as like trial number?
Yes, yes, yes. It's like the stimulus presentation number. So the stimulus was presented once, stimulus presented the second time, the third time. It's just trial number. Yeah. Okay, so the next thing that you can do from a dot raster is make a post-stimulus histogram. And as the name suggests, the post-stimulus histogram is basically just telling you, okay, we presented the stimulus, and after that we want to see what the behavior of the neuron looked like. The difference between this and mean firing rate, well, the difference between this and the dot raster is essentially that now, yes, you're looking at the time after the stimulus was presented, but you're taking all of that data of when the time points of when the neuron fired, you're taking that data and you're trying to find firing rate. And so you're trying to figure out, maybe you're trying to figure out after the stimulus was presented, at what how long does it take after the stimulus was presented to get the most firing rates for this neuron to be firing the most. Any questions about that? Yeah.
When you say calculate the number of spikes within bin, is that the number of spikes per neuron per trial?
Yeah, so that data from the dot raster there is all just one neuron. So we're just looking at one neuron. We're presenting the stimulus to it multiple times. So there are multiple trials. And so what we're trying to now look at is, okay, so we generally kind of have an idea of after the stimulus is presented, how does this particular neuron tend to behave? across multiple trials. And so now we can, because we did it over multiple trials, we can now sort of find a mean firing rate. So for what we do for that is we define a bin width. So our bin width here might be 10 milliseconds. And so for each 10 millisecond after the stimulus was presented, we basically just we count the number of dots within, so sort of the red box that I've made on the dot raster. We just basically count the number of spikes within that red box, and we divide that by the number of trials, and divide that by the bin width, so by 10 millisecond, to get what was the mean firing rate just within sort of that bin width. So within sort of, let's say, 70 milliseconds after the stimulus was presented and 80 milliseconds after the stimulus was presented. So at that sort of time interval, what kind of firing rate did we get over multiple trials? Yes?
I think in one of the questions from Juan's packet, there was a situation where we're given a stimulus that is presented for some period of time, but the, I guess, time points for firing that he gave began at a point before the stimulus was presented and extended to a point sometime after. Mm-hmm. In that case, should we include the whole time window or just the duration where the stimulus was there?
Well, this is a post-stimulus histogram, so what you would do in this case, and the TS can correct me if I'm wrong, what you would do in this case is to look at sort of after the stimulus has started.
Like 5 time points after the stimulus ends. So
After the stimulus ends?
yeah.
Do you guys have an answer to that? Which exam was this on?
Yeah, it's question 2 for 2017.
Question 2 for 2017. Can someone look into that?
Or at least the both thing,
A sensory neuron is stimulated by a 100-foot sinusoidal stimulus starting at 100 milliseconds. Below is a list of the time of occurrence of spikes report from the neuron in response to 5 repetitions of the same stimulus. Recordings begin at time equals 0 and end at time equals 200.
Yeah, so I would assume it's, when it says post-stimulus, it means since the beginning of the stimulus. Yeah. That would be my guess.
Yeah.
Is there a convention for when like a specific point should be included in an event? Like, so in the 2016 test, there's like an example of like you're given all these points and like some values are like 90, 100 that would be on like the edge of the bin widths. Like, do those go in the lower bin or the higher bin? Because like that changes the histogram.
For that, I would say just be consistent with what you do. So whether you put it in the lower or the higher one, and indicate what you did. So we'll give you points for that. That's just a small technicality. Any other questions? Yeah.
Also, in that question, it then asks if there's phase locking in the neuron, but like I don't know how we would, I just don't know how we would tell that from the post-stimulus histogram.
So we'll get into that on the next slide of period histogram, because period histogram is actually very similar to a post-stimulus histogram, because you're still measuring firing rate. So let me go over period histogram. If you still have questions on phase locking, we can talk about that. Any other questions? Yeah.
What do the y-axis say?
So the y-axis here says firing rate, and that's the reason why essentially to calculate that you would go across the different trials in the dot raster, and you would count all of the spikes, so all of the dots, so all of the action potentials, and you would divide that by the number of trials that you've looked at and by the bin width, so maybe 10 millisecond if that's your bin width. Any other questions? We'll move forward. Okay. So what we have next is a period histogram. And so this is actually similar to a post-stimulus histogram. The difference here is that now you have a periodic stimulus. And so in a post-stimulus histogram, what we had was the dot raster, where we had, we have a particular neuron, and we're presenting a stimulus to it multiple times, and as a result, we're able to make sort of different, we're able to sort of have different trials laid on top of each other, which gives us sort of some data to work off of. Now, in our period histogram, we're not doing multiple trials anymore because we have a periodic stimulus. And so periodic stimulus is actually very similar to multiple trial experiment because it's periodic stimulus in itself means it's a stimulus that repeats itself. And so what you need to understand there is that you essentially are doing the same processing as a post-stimulus histogram because it is a multiple trial experiment, except the different trials are now segments of the periodic stimulus. And so if we just set an arbitrary 6 periods to be one segment, so that's what Dr. Wang does in his lecture. If we set that to be one segment, then that's almost like one trial. And when we create a dot raster for this particular neuron, which is responding to a periodic stimulus, we create the dot raster keeping sort of each segment as a trial. And so each segment is basically just a part, so maybe 6 time periods of this periodic stimulus. Does that make sense? So what we have here, just gonna get the pointer. Okay, what we have here is, in order to interpret everything that's happening in this diagram, so I've tried to annotate it a little bit to make it clearer. So if we just look at this 768 Hertz stimulus, so this is a periodic stimulus. The way Dr. Wang split it up is he assumed, okay, let's let 6 periods be one segment, and so you essentially end up having 3 segments, and so that basically counts as 3 trials, essentially, when we're creating our dot raster. And so then we look at, along those 3 segments, we look at the firing of this neuron. And so that's just the action potentials of the neuron, which he's basically split off into the 3 segments, so the action potential corresponding to the 3 segments. And accordingly, he was able to sort of make a dot raster. And so the dot raster here, what you have to recognize, is pretty much the same as the dot raster we've had all along. The only difference being, instead of different trial numbers, we have different segment numbers. And so we're looking at 3 segments instead of 3 trials. But we are still looking at sort of the same x-axis of time after the stimulus began, or after the segment began. So we're still looking at time on the x-axis. It's just that on the y-axis, instead of trial, we're now looking at segment number. And so what we do now is the same idea as a post-stimulus histogram. Is what we do is we look at sort of the time points that are below that dot on the x-axis of that dot raster, and we make again a stimulus a histogram wherein we've defined a segment length of n periods, let's say 6 periods. That allowed us to make our dot raster. Now we define a certain bin width, so we might define our bin width to be somewhat less than, maybe we define it to be 0.5 millisecond. And so for every 0.5 millisecond, we're still calculating firing rate, except now we're calculating firing rate across different segments and not across different trials. And that's how we develop our period histogram. And phase locking can now be determined from the period histogram, because what we see here is that the 1.3 millisecond period of the stimulus matches the 1.3 millisecond time interval between two large peaks of the sort of firing rate of this particular neuron. What this essentially tells you is that you have a certain stimulus, which is sort of I don't want to say firing, but like the stimulus is sort of periodic with a certain time interval. Right? And what you have with your neuron, what you've calculated now with your neuron, is that largely it is firing at a time interval that is the same as the time interval of your stimulus. And so it's almost like saying that your neuron is in sync with your stimulus, with your periodic stimulus. So the way that the analogy that I make in my head is that if you have if you have music playing and there are certain beats in the music which are like periodic beats, and you're sort of like tapping your foot along with those beats, your foot is in sync with the beats of the music. And so similarly, when you have a stimulus with a certain time interval, it's periodically sort of that stimulus is playing. Your neuron is also in sync with that stimulus and is firing largely in sync with that stimulus. Any questions? So phase locking, actually, there was a Piazza question on this. Phase locking, your auditory nerve will be phase locked. Your cochlear nucleus, so sort of like with higher levels of processing of that same information as you go above in the auditory pathway, and we'll go over that next. So we'll cover this again, but as you go higher in the processing, the auditory processing, you lose some of that phase locking. So it's still there, but it's less and less in sync because you're going higher up in the brain. Any other questions? Yeah.
The x-axis of the period history.
Oh, it's just time interval. So it's basically just, it's the same as the post-stimulus histogram, wherein you're just looking at the time after the segment began. So each of these represents a different segment, right? And so this is sort of the firing after that particular segment began. So after segment 1 began, or after segment 2, or after segment 3. And so you're just looking at the time across this particular segment. That's what this essentially will end up representing. Yeah.
So if you have the neuron firing as like a harmonic of the stimulus, so that like if it's exactly twice or exactly 4 times or something, is that still phase lock? If it's firing every 3 stimuli?
Do we have an answer to that? Any of the TAs? Is it still phase locked if it's like it's twice the time interval or thrice the time interval? What does it have to be exactly?
So if the firing rate of the neuron is like some exact number of stimuli,
Right.
so it's exactly 2 stimuli or exactly 3 stimuli, would you call that phase lock?
So you're asking if sort of the 1.3 millisecond here doesn't match the 1.3, if you have sort of 2.6 instead.
Yes.
So
I feel like
the idea with phase locking is not that the neuron will fire once every cycle. It's just that when it does fire, it kind of fires at the same points in the cycle. So my intuition is to say yes, that's still phase locking. It's just that instead of phase locking to a single point in each cycle, it's maybe phase locking to a couple of points. Does that make sense?
Yeah. Also to add to that, that would make sense if it were still sort of 2.6 or 3.9 or something. It would still work out, because the reason you need phase locking is because of binaural processing. So you have sort of, when you talk, your voice goes to one ear of mine and to the other ear. The reason we need phase locking is because we want the auditory neurons to respond to the stimulus at sort of in sync to the stimulus, so that they're in sync with each other, so that when they meet within the processing of the sort of, it meets before the brain, we'll go over that, but when the sort of the auditory nerve firing sort of meet each other, we want them to not miss each other. That's why you need them to be synced with each other, so you sync them to the stimulus. So as long as both auditory neurons were, like in the right and the left ear, were still synced with the neuron, even if it were sort of at a 2.6 millisecond or a 3.9 millisecond, it would still work out, because the signal would not miss each other. The two signals would not miss. Yeah.
I just wanted to clarify for the bottom graph, the period histogram, when you define bins, each bin would have to be significantly smaller than the period of the stimulus itself.
So in this case, you would have sort of a bin width of, so if we have, yes, so if we have, so we're looking at 1.3 millisecond is sort of the period of the stimulus, right? And we're looking at 6 periods. And so that's 6 times 1.3, let's say 6 milliseconds or 7 milliseconds. So you're looking at 7 milliseconds total. And we know that along that sort of along those 7 milliseconds, which are sort of represented over here, which is why it says 5 over here. So you have 7 milliseconds in total over here. You would have somewhat firing across maybe every 2 milliseconds. You could count sort of 3 dots, or as opposed to that 1 dot. And so in this case, the bin width that I would go with would be about 2 milliseconds.
Just one follow-up, sorry.
Mm-hmm.
Just because, like, locking, like the spikes are matched with each stimulus at the same point. So wouldn't you need like within one period of the stimulus, like a bunch of bins within that to count all of the action?
So this dot raster doesn't accurately represent what, so this is a very simplified dot raster. What the dot raster would typically look like is closer to the dot raster that we've seen over here. And so they wouldn't necessarily be aligned with each other. And so you should be able to have sort of a 2 millisecond interval that you call your bin width, within which you have some amount of firing in segment 1. They're not sort of totally aligned with each other. So this is just sort of, I wouldn't consider this to be an accurate representation of what the dot raster will look like. The dot raster will look more like this, wherein they won't be aligned with each other, directly aligned with each other. Yeah. Yeah.
Matters that there's like 6 different stimuli, because it seems as though we're only looking at one like stimulus.
We're just looking at one.
So the fact that it like fires multiple times, like we don't care about that at all.
No, I would not go into that, because you want to see the stimulus, the periodic stimulus, as a combination of segments. And over here, instead of trials, you have those segments. We're not going to go into like 3D processing of like, okay, what if you have multiple periodic stimuli, because then you have segments and you have multiple trials. We're not going to go into that. No. Yeah.
Sorry. So the top row is.
So this one or this
What
one?
above it?
Sorry?
The very top row of the diagram where it's like.
This? Okay, so this is the stimulus. So this is a periodic stimulus, just that little black thing.
Yeah.
And then these are the spikes associated. And so this whole diagram is basically just a blow up of what you're seeing over here.
Yeah.
Yeah.
But are you looking like a segment of time contains multiple stimuli delivered, or like how are you defining like the segments based on like the period of delivering the stimulus, or like periods within the stimulus itself?
Periods, so 6 periods within the periodic stimulus itself. So we're not looking at multiple periodic stimuli given in the experiment. We're just giving one periodic stimuli. So this is literally your stimuli, just this one 768 hertz tone that you've given.
Okay. And then how are you defining your bin width again? Since if your bin width is 2 milliseconds and the period of like the stimuli itself is like 1.3 milliseconds, wouldn't like all the firing from one stimuli be within that bin?
Okay, yeah, you're right. I would do it smaller than that. I would do it along 0.5. And essentially what the dot raster would hopefully look like would be, you would have more dots on it, so you would be able to, within that 0.5, sort of have, okay, within this tiny little segment here, what was the firing rate? And so you would be able to make sort of a histogram box that goes like that, and then a histogram box that goes like that, and then maybe one that goes much higher that shows, okay, at that particular time point, within that time interval, you had a lot more firing. Yeah.
So I guess back to the question I asked earlier. So would basically, if it has phase locking, it would just look like multiple spikes, not like a curve like most like post-stimulus histograms we've seen would look like?
So over here, what you have essentially is one stimulus being presented. It's not a periodic stimulus. It's just one stimulus was presented, and you're looking at this neuron's behavior after that stimulus was presented. And so what you would expect to see is that for some time, so over here, you don't really have that much firing because it needs a little bit of time to respond to the stimulus, and then all of a sudden you have really high firing, and then that firing kind of attenuates over time, which is why you don't have multiple peaks here. But in a period histogram, you have a periodic stimulus, and so the neuron is kind of required to fire multiple times since the beginning of the segment. So since the beginning of those 6 arbitrary periods that we've defined within our periodic stimulus.
But it asks for like us to make a post-stimulus histogram of like in question 2 of 2017.
Okay.
For a sinus
sinusoidal stimulus, so that it would be a periodic stimulus, but it's asking us not to do a period histogram specifically, but instead to do post-stimulus histogram.
And then it's giving you time points, I imagine,
Yeah.
of when it fires. I would just, in that case, I would not complicate it any further. I would just make a post-stimulus histogram of, you're assuming that it is a single stimulus that's been given to you. You're not looking at how the neuron fires along with that periodic stimulus. You're just, it's almost like you're giving the stimulus and then you're taking away the stimulus and you're looking, okay, how does the neuron keep firing after the stimulus has been given once?
And so how would it, if we were to do the post-stimulus histogram for this periodic stimulus, you would say that there's phase locking if it looks like distinct peaks, sort of like the period histogram, or something else?
No, to see phase locking, you always need to be able to see that there are multiple peaks. And then those peaks represent largely the time interval between two large firings, between two, sort of the time interval between two major firings of this neuron, and then that should sort of match the time period of the stimulus, which is 1 over the frequency of the stimulus. Any other questions? Okay, in that case, we'll move on. So now we're making sort of a ISI histogram from the dot raster, and ISI histogram is basically interspike interval histogram. So the difference here is that we're looking not at the time. So the x-axis here is not the time after which the stimulus has been presented. So we're not just looking at, okay, for the next 600 milliseconds after the stimulus was represented, let's look at the behavior of the neuron. We're not doing that anymore. We're now looking at interspike intervals. So now we're trying to see how far apart are the firings of this neuron after the stimulus has been presented. And so what we do for this is we have, on the x-axis, we have interspike interval. And this would also be created from a dot raster. And so the dot raster basically gives you information about, okay, within a particular trial, once you've presented the stimulus, you have this neuron sort of, responding, firing after the stimulus has been presented, and maybe at a certain, maybe the interspike interval of that particular neuron is most likely to be 10 millisecond after the stimulus has been presented. And so what you're doing now is you're defining ISI bin width. What I would do is calculate first the ISI interval, so the T2 minus T1, T2 being sort of the second firing and T1 being the first one. So calculating the sort of the time interval between each firing out of the different time intervals that have been given to you in the question. Then I would calculate the percent of ISIs, so the number of ISIs that fall within a certain ISI bin. So once you've decided on a ISI bin width, you would just look at, okay, out of the ISIs that I've calculated from the data that was given to me about this neuron's behavior, I would look at how many of the ones that I've calculated fall within a certain bin width, and that allows you to make an interspike interval histogram. And what's important to notice here is that the x-axis is interspike interval, but the y-axis is percent of ISI. So it's almost like the probability, it's almost a probability measure, because you're saying the percent of ISIs that were found to lie within a specific ISI bin. So for example, between 10 millisecond and 12 millisecond. So how likely is the neuron's ISI to be between 10 millisecond and 12 millisecond? And so this is almost a probability measure, the y-axis here, because we're calculating it in terms of percent. And so what we can do now, since this is a probability, sorry, do you have a question?
Yeah, can you go back to the last page? Why is it specifically a probability?
Oh, the reason is just, we're just defining our y-axis as a percentage. So the percentage of ISIs that, out of the ISIs that you've calculated from the data given to you about this neuron's behavior, you're looking at the percentage of ISIs that lie within a specific bin width that you've, out of that entire bin width, out of the bin width that you've decided for the entire time period that you're looking at. So maybe you're saying, okay, by 100 millisecond is kind of like spontaneous firing. And so after the stimulus was presented, the chances of having 100 millisecond apart action potentials, quite unlikely. because mostly the ISIs that you calculated were closer to 10 millisecond and 12 millisecond. Any other questions? Okay. So basically now, since this is a probability measure, it is possible for us to basically fit this curve with a Poisson function or with a gamma distribution. And so the difference between using Poisson and gamma would essentially be that in Poisson it would look like that, wherein you're saying, okay, at 0 ISI, I have this really high probability of sort of the high probability of my ISIs falling within that bin width of 0 to something. That is kind of misleading because you can't really have the ISI ever really being 0 because every action potential requires a refractory period, which is why the Poisson isn't a great way to model the sort of the interspike interval histogram, which is why the gamma distribution is kind of better, because the gamma distribution allows you to sort of have at 0 seconds, so at 0 ISI, you still have a 0 probability of having your ISI sort of fall within 0 millisecond bin. Yes?
So on the homework, we were also able to do with the refractory period using like a shift.
Right.
Temporal shift. Is there like which way using like a temporal shift of a Poisson using a gamma versus a gamma distribution?
Either is fine. Yeah. And so yeah, so the advantage of gamma when compared to this Poisson is basically just that it allows you to account for dead time or refractory period. And then the bump at the end of the graph. So if there is a bump at the end of the graph here at 0.06 or at any other time point, that basically shows phase locking. And the reason for that is because you're saying that every 0.0 seconds, there seems to be some amount of increased firing. And so this, it's almost like this neuron is firing periodically and is kind of wanting to fire again every time at 0.06 seconds, at an interval of 0.06 seconds. And so that shows phase locking, and the same bump could exist in a gamma distribution as well. And so if you're asked, you know, how would you see phase locking in an ISI histogram, you just want to be able to indicate that it's really the bump that shows you phase locking. Or if you're told to interpret this ISI histogram, you could say that there is phase locking. Yes?
So is there a quantitative way to say that? Like, does it have to be a certain type? Or it could be
No, not really. No, this is highly qualitative. You won't be asked to go into that much detail. No. Okay, so now let's talk about some auditory biology and sort of tackle the question of how is sound converted into neural firing. And so the level of depth that I recommend knowing for this is basically the amount that Dr. Wang goes into in his lectures, not necessarily further than that. But you want to know the amount that he goes into, which is basically to say, okay, you have sound coming into your cartilaginous area of the ear, which is basically just your pinna. So that's what collects the sound vibrations. It goes through the ear canal, and it then hits the eardrum or the tympanic membrane. And so you have vibrations hitting the tympanic membrane, and this leads to sort of some downstream things that happen, which we'll go into next. And so what happens now after the tympanic membrane is now vibrating as a result of that sound vibration is you have 3 bones called the malleus, incus, and stapes. All 3 of those sort of tend to bang on each other, and so they sort of move each other in sort of like a signaling cascade. They move each other, and what you have next is that it sort of bangs onto the oval window, which is basically sort of the window that leads into the cochlea. And so that's how basically the sound vibrations are transmitted all the way to the cochlea. And so let's talk about the cochlea now. The cochlea is important because it's sort of this circular structure, the spiral-like structure. And sort of this might be the oval window. And so what we have running all along the cochlea is the organ of Corti. And so the organ of Corti runs all along the cochlea right in the middle. What you have on the sides of the organ of Corti are 3 different sort of empty cavities which are filled with fluid. And so as you can imagine, every time the malleus hits the incus, hits the stapes, which hits the oval window, there is some amount of vibration in the fluid that is in the cavities, the 3 cavities of the oval window. And so that's kind of important because it moves the organ of Corti. And so what does the organ of Corti consist of? The organ of Corti has, yep, that's a good representation of it. You have basically a basilar membrane at the bottom. You have a tectorial membrane on the top. And in between, you have inner hair cells and outer hair cells. So you have three rows of outer hair cells running all along the cochlea and one layer of inner hair cell running along the cochlea. And so the outer hair cells connect with the afferent nerves. Afferent is sort of the arriving signal, and efferent is the exiting signal. And so you do have some signals that go from your brain to your ear, and so those would connect with your outer hair cells. Whereas the afferent signal, so the arriving signals, the signal that arrives from outside, will sort of will be transmitted through the inner hair cells. And how does that transmission happen? It happens because of the vibration in the fluid in the cavities of the cochlea. And why does that vibration happen? Because the malleus incus stapes are sort of banging on the oval window, because prior to that, the tympanic membrane is having vibration as a result of sound vibrations. Question?
Sorry. So the inner hair cells are the ones that respond to the afferent signals?
The inner hair cells, yes. The three layers of inner hair cells respond to afferent signals. So the sound that you're hearing from outside.
Three layers of inner hair cells?
Yes, so 3 rows of them. So you have, as you can see over here, you have 3 rows of them, as opposed to the 1 row of outer hair cells.
Wait, isn't that inner and that's outer?
Sorry, did I make a mistake? Okay, this is the outer, which is 3 rows, and the outer is the... Okay, maybe I made a mistake here. Let me check. Maybe I made a mistake. But what's important to know is the process by which this transmission happens, which is the fluid vibrations basically leads to the movement of the tectorial of the basilar membrane. So the basilar membrane moves up or it moves down as a result of the vibration of the fluid inside the cavities. And when that happens, your hair cells, the inner hair cells and the outer hair cells, sort of brush against the tectorial membrane, and it's that shear stress that is experienced by the hair cells. And so something that's important to know now is that the hair cells have stereocilia on top of them, and so they have hair on top of them. That's why they're called hair cells. It's really that hair that sort of bends as a result of that shear force, which leads to depolarization of the hair cell. And so that depolarization of the hair cell as a result of the shear stress that's experienced because it's brushing past the tectorial membrane because of the vibration in the fluid cavity and the movement of the basilar membrane, as a result of that depolarization of the hair cells, we now have a signal transmitted to the auditory nerves that are connected to the hair cells. And so this is basically just a summary slide that collects everything that I just said. And so you can go over that afterwards. Yes?
So basically for this slide, we just kind of need to know the sequence of it rather than
Yeah.
labeling of any...
Definitely, yeah. You want to know the sequence of it. Yeah.
So the outer hair cells are connected to the efferent nerves. Those are sending the signals to the brain, right?
Yeah, so the way I always memorized it is efferent means exiting signal. Afferent means arriving signal, and so afferent signals are the ones that are coming from outside, and so like your voice is an afferent signal into sort of my auditory system, as opposed to efferent signals are signals that are going from the brain to the auditory system, to the ear.
So what are the inner hair cells doing the position?
So you mean why would there be a need for efferent signals? For the afferent signal, so those are the ones that are transmitting your sound vibration as sort of that's what leads to your sound vibration being converted into auditory nerve firing, which is then taken to the brain to be processed as sound. Yeah.
I thought efferent signals are sensory. They go from the inner hair cell to the auditory.
Okay, let me check on that. I might have made a mistake. Can the TAs correct me in case I've gotten the efferent and the afferent mixed up?
Pretty sure afferent is to the brain and
Afferent is to the brain and efferent is from the brain. That's what I agree with. Yeah.
Would we be expected to like draw a structure or label a structure as well?
Draw, no, because he's made it clear in his lectures that he just wants you to know what's what, and usually puts a red box around what he wants you to know. Yeah, so I would prepare for labeling. I would not prepare for drawing. Yeah. Okay, and so how is frequency selectivity generated? So in his slides, he basically goes over 3 large questions, and so the first one is what we just covered, how is sound converted into neural firing, and the second one is how is frequency selectivity generated? You want to know the answers, like the large answers to these questions, because those are kind of the guiding questions in his lectures. And the way he usually likes to test as well. So how is frequency selectivity generated? And so essentially what we have in the cochlea, so cochlea is this spiral structure, which we now, if we sort of open it out, we sort of have this. Sort of once we open it out, we have the apex of the cochlea, and we have the base of the cochlea. And the important thing to note here is that as you move from the base to the apex, the selectivity, the frequency selectivity of the neurons changes. And so the neurons that are connected, the auditory nerve fibers that are connected to the apex of the cochlea are most sensitive to low frequency sounds, as opposed to the auditory nerves that are connected to the base of the cochlea, them being more sensitive to high frequency sounds. And so the way that and all along the rest of the cochlea, from the base to the apex, you do have sort of this tonotopic organization of sort of progressively, progressive sort of decrease in progressive change of selectivity towards certain frequencies of sound. And the way that I always saw it is almost like it's a flag. So if you had a flag and it was like a not windy day, maybe there was a little bit of wind, you would have, maybe the apex of the flag would still be moving, but the base of it wouldn't really be affected, as opposed to if it was a really windy day, where even the base of the flag would be moving. That's kind of just an analogy that I used to memorize this last year. But what you just want to be able to understand is that the apex is most sensitive to low frequency sounds, the base is most sensitive to high frequency sounds. And you can relate this back to sort of an audiogram, in that if you were to take the audiogram of the neuron, of the auditory nerve that's at the apex, and so that's in this case C. So as you can see, C is most sensitive to 1 kilohertz frequency sounds, as opposed to A, which is a neuron at the base of the cochlea, which is most sensitive to 10 kilohertz sound. Any questions on that? And so this is basically the tonotopic organization of the cochlea. And so now what we have is pathway from the cochlea to the brain. This tonotopic organization is maintained. And so the different frequency selectivities is sort of maintained all the way. So you have this in the cochlear nucleus, superior olive, where binaural processing takes place of ipsilateral. Ipsilateral basically just means from the same ear. Contralateral means from the opposite ear. Inferior colliculus goes to thalamus, goes to auditory cortex. And what's important to know is that in the auditory cortex, this tonotopic organization is maintained. So as you can see, when you have a certain, maybe my voice, that sort of stimulus where there are multiple frequencies within that sound, you would have a sort of corresponding frequencies. With the corresponding frequencies, you would have the corresponding auditory neurons being fired. And so those are the neurons that are responding in that image right there. And so the third question that he goes over is, how is time represented by neuronal firing? And so the answer to this lies in phase locking. So phase locking of a periodic sound stimulus and the auditory nerve firing preserves the time representation of the stimulus. And so the phase locking is maintained in cochlear nucleus and in the auditory cortex, but the strength of the phase locking is reduced with higher levels of processing. And so that's what this diagram right here shows, that as you sort of move towards from the auditory nerve to the cochlear nucleus to the auditory cortex, you have, for a certain frequency of sound, you have sort of less phase locking. And yeah, any questions? Yes?
Is there a mechanism behind why it gets progressively worse as you go up
Not that I know of. Do any other TAs have an answer to that? Why does phase locking reduce with higher levels of processing? No? Yeah.
I think it's because, like, at your earlier levels of processing, like after you
like
Yeah.
when it initially hits your ear, you like need a stimulus.
Right.
You like coded properly, and that's why you're phase locking.
Also because by then you've crossed the binaural processing stage. So binaural processing is where you really need the phase locking because you need the signals to meet each other. They can't miss each other. So one goes first and then the other one goes from both your ears. And so they need to meet. And so in order to meet, they need to be synced with each other. And so once you've sort of reached that state in at your superior olive, you don't really need the phase locking after that. And so yeah, that's what I have. Okay. Yes, questions?
It asks in one thing, but like it for the source of phase locking, would that just be like why it like to keep like binaural processing, or is there anything like that?
So the source of phase locking would be the periodic stimulus.
Okay.
Yeah. That's the answer that I would give to that. The reason for phase locking, the significance of it would be the binaural processing, but the source would still be the periodic stimulus. Yeah. Any other questions? Or we'll move
I was
on.
wondering if you could go back to the last slide for like 30 seconds. Okay, yeah, I just wanted to write some stuff down.
Yeah, I'll post these on Blackboard.
Okay.
Yeah. Okay, yes.
On the diagrams of the processing levels, some of the labels have like red dots next to them. Do we need to memorize those?
You just need to know what's on here. So I just collected whatever he put a red box. Yeah, so you want to know the ones that he put a red dot next to. You're right, yeah. You should know those. Yeah, any other questions? Yes.
In the slide after this, on the time one,
Yeah.
what was the best frequency on the x-axis?
Characteristic frequency. And so that's the frequency that, okay, so this basically goes back to the point of, so you have phase locking in response to periodic stimulus, right? And so this periodic stimulus has a certain frequency, right? Now, the thing with phase locking is that it's better at low frequency sounds than it is at high frequency sounds. Because as you can imagine, at really high frequency sounds, your neuron would have to fire really fast. And it can't necessarily do that because it needs to have a refractory period. And so what this basically shows you is it gives you two pieces of information. One is that there's less phase locking at higher levels of processing, but also the fact that for a single, let's consider the cochlear nucleus, at lower levels of frequency of sound, you do have higher phase locking than at higher levels of frequency of sound. Yeah.
So you said that like the phase locking is not really shown in the brain, like it was like the strength of phase locking is maintained in the auditory cortex, which like on the last page.
Oh yeah. So it is maintained. It's just that it's to a lesser degree. Yeah, it's just not as much phase locked. And so the strength of phase locking is determined by the synchronization index. As the higher the synchronization index, the more the phase locking. And so that's what this diagram shows. That you do have some amount of phase locking all throughout your auditory system, but with higher levels of processing, it decreases. Yeah.
What is the synchronization index and like how do you
Calculate it. He has a calculation for it on his slides.
Okay.
Yeah. I can show it to you afterwards, but in the interest of time, I really want to move to the next professor, since you have like 7 professors. Yeah. Okay. Dahlia.
That might be good, yeah.
Okay, got it.
Okay. Do I have to press anything for the mic, or is it already on?
I think you should be fine. Can I check on it?
Yeah.
It's on.
It's on. Great. Oh, right. Okay. Can you guys see this? Is this okay? All right. Okay, so we're going to talk about Winslow. Yeah. Lights on. Is that better, worse, better? Okay. Okay, so Winslow went over a bunch of things. We're going to start with cable theory. And in general, for Winslow, even though the derivations are good to know, like good to know where they came from, that kind of thing, I would worry more about being able to solve like the cable theory problems he gives you, so more similar to like the practice problems you've seen on your homework, than like knowing every step of the derivation for cable theory, because I think like also Trianova has done cable theory before, so like should be pretty familiar with that, and that's like not the main point of what Winslow is doing. So I'm just going to go over a couple practice problems, and the overall strategy for the problems is that usually you're given the outward conductance, the G out of the ends of your branches, and you basically want to start with those knowns and then move inwards to get the G ins using those equations, and then the G out of like the branch where you're inputting the current. So you're basically just kind of like iterating backwards. So the most important equations to know are the like the G in equations. So G in is equal to G infinity and then times G out over G infinity. And then so it's going to save you a lot of time if you just memorize these equations. So I would definitely recommend that. Those are like the three main ones to know. So starting with this example, you're going to have, so all of these branches are going to be equal length. They're all length L. This is branch 1, branch 2, branch 3. You're going to have a current input at the end of branch 2. And they all also have the same G infinity. And then one example of a question would be, these are questions that you've seen on your homeworks, are what is the voltage attenuation at the branch point relative to the input point? So what that is basically asking is what is V1 over V2? So the overall strategy is, again, to start with these G out values, and for simplicity here they're 0, and then work backwards, because in order to get what V1 is, we need to know what this G out is here. So first we're going to try to find G out 2. So that's step 1. And then if you notice that, so conductances add, since these are basically like in parallel. So this G out 2 here is going to be equal to the G in 3 plus the G in 1. And then using your equation for G in, for example, G in 1 is G infinity times G out over G infinity plus tanh L over 1 plus G out over G infinity times hyperbolic tangent of L, and G out is 0. So that basically means that your G in 1 simplifies to G infinity times tanh L. And then, yeah. Oh, it's okay. Are the like sideboards really hard to see for you guys? Okay. So this just says step 1. Is this better? Yes. Okay. Sorry about that. Step 1 is to find G out 2, and that's just equal to G in 1 plus G in 3. And then using your equation that you memorized, G in 1, and also using the fact that G out of branch 1 is 0, which is given to you. If they give you a different G out value, you just plug in that value, but it's simpler if it's 0. In this case, that's just G infinity times hyperbolic tangent of L. So that's, and then you can notice that since the problem gave you that they're all the same length, and the G infinity, which is like a property of that particular cable, are the same, branch 1 and branch 3 are essentially identical. So you can notice that these are then equal to each other. So G in 3 is equal to G in 1. If that weren't the case, if they gave you different lengths, different G infinity values, whatever, you would have to recalculate. So then you basically get that G out 2 is equal to 2 times G infinity tanh L. So now we have the G out of the branch that we're looking at, which is branch 2. So now you can get the ratio of V1 over V2 using the V of x over V of 0 expression that's up there. So V1 over V2 is V at L, because the length is L, over V2, which is, so this is cosh L minus L, so that's 0, plus 2 times G infinity tanh L over G infinity sinh of 0, L minus L, all over hyperbolic cosine of L plus, again, 2 G infinity which basically just simplifies to 1 over cosh L plus 2, sorry, I'll move out of the way, which basically simplifies to this. And that's going to be less than 1. So then the next question would be, what is V0 over V1? So that's like, what is the voltage attenuation at like the origin of branch 1 over the branch point? So that's question 2. V0 over V1. Sorry. It's v0 over v2. So what's the, if v0 is the soma, then like what's the attenuation at the soma versus the input point? So for this type of question where it asks like multiple branches, you want to go branch by branch. So you can notice that v0 over v2 is the same thing. Can you guys see down here as well? Or is that too low? Yeah, okay. v0 over v2 is v0 over v1 times v1 over v2. The v1s cancel. So we have v1 over v2 from the first question, question A. So now we just need to get, yes?
In that last part, what happened to v2? Like, why did we not have to calculate anything for that?
Oh, okay. So v2, the question is like, what is the voltage attenuation? If that's like the wording of the question, I guess I didn't state that quite so well. So that's basically asking like, what's the ratio of v1 over v2? But if you were going to calculate v2,
is that equal to v1 0?
v1 0.
0, because like the equation up there for vx is.
Oh, yeah.
Okay.
Yeah. Yeah. Alternately, you could also notice that you could calculate the input, you have the input conductance, and then V of 0 is the current, which they would give you, like if they asked you to calculate that, they would give you the value of I2, or you could just use I2 and plug that into V equals I over G. I think if that's right, yeah, yeah.
For branches 1 and 3, are you choosing 0 to be where V1 is and L to be at the end?
Sorry?
How do you choose where 0 and L are for branches 1 and 3?
Where 0 and L are for branch 1 and 3. Oh, okay. So it depends on the, so like, okay, so if you're calculating, the question is like, what is V1 over V2? Then your origin is here, so that's 0, and then this is L, because this is like the length L. And then if you're starting from here and calculating what V3 over V1 is, this is going to be 0, that's going to be L. Does that make sense?
Always follow the current.
Yeah.
Where the current goes in, that's where the 0 goes.
Yeah. Yeah. And then if it asks you like, what is the voltage here, which is like at L over 2, you would still calculate this G out, but then for your X here, the voltage V of X over V of 0, you would plug in L over 2 instead of L. Yeah. Okay, so now to get, yes, yes, where? Sorry. Yes.
Whenever a question states that like there's sealed in boundary conditions at all non-branching ends, does that mean every end that isn't the branching point?
Yeah, so anywhere, wherever you're not inputting current and wherever you're not branching is, so like here, at the point 0, V0, where the V0 label is, that would be conductance is 0, the sealed end boundary condition. Where the point V3 is, that would also be 0, but then where the point V1 is, that's a branch, so you cannot assume that the conductance there is 0, because otherwise, like, you wouldn't have any current flowing through. Yeah.
Just to check, if you are trying to find like the calculations at L over 2, can you still apply the current law, like the additive current law, the junction rule?
The junction rule?
Like the G out 2 equals G in 1 plus G in 3.
Yeah.
Does that still apply?
Yeah.
Okay.
Because you're still calculating the G out, like, at the branch point. Okay, so any other questions for, okay. So the next, so we have V1 over V2. So now we're looking for V0 over V1. So we can notice that G out at V0 is 0 from the sealed end condition. And then the distance at which we evaluate V0 is L, because we already were told that the length of branch 1 is L. So then you can just plug in, for here, V of X over V0 is equal to hyperbolic cosine of L minus L, so hyperbolic cosine of 0, which is just 1, plus G out, which is 0, over G infinity times sinh of L minus L, so that goes to 0 since G out is 0. So that's 1 plus 0 over hyperbolic cosine of L. So plus another expression involving G out, which is 0. So it's just 1 over hyperbolic cosine of L. So now you have V0 over V1 and V1 over V2 from our answer here, and then you can just multiply them together and get V0 over V2. So basically you're just going branch by branch. Let's see. Another question they could ask, is what is V3 over V2? And you could go through and do the whole calculation again, but it's easier to notice that since we were told that V3 and V1, like all of the branches have the same length L, they all have the same G infinity, going from here down this path is the same thing as going from here down this path. So V3 over V2 is the same thing as V0 over V2. So make sure you look for patterns like that. That'll save you a lot of time. Similarly, V3 over V1 is the same thing as V0 over V1. So if it asks you what is V3 over V1 and you've already calculated V0 over V1, you don't need to recalculate. Unless, of course, you have a different length or a different cable property.
Same for the situation, V3 over V2 is the same as V0 over V2?
Yes, for this particular problem. But if it doesn't, like you can't assume that in general. It's only because the properties of the cable are the same throughout, and that was in the problem statement. Okay. Okay, so I don't think he didn't cover he mentioned the and not operator, but he didn't like go through it, right? Is that correct? Assuming, yes, okay, so we're gonna go over the and not operator. And it's like a slightly more complicated problem, but you just kind of have to break it down, and it's mostly just like annoying computation. It's not necessarily super hard, but you just have to like have a general strategy of how to approach it. And then any kind of related problem that he gives you or might give you, like you just have to break it down. So this is okay, so this is the structure of the and not operator. So this is basically, this is your excitatory input. So this is like EE is like a battery, and it has some non-zero voltage value. And you're gonna have a conductance, GE. And once you turn this on, or like once you turn the conductance on basically, you're gonna have some current flowing through here. This is the inhibitory input, and basically what this is like this EI value is 0, or like equal to ground, which we're gonna say is 0. And then once you turn that conductance GI on, it's basically gonna shunt current away. So like imagine your goal is to get current to this point here at length L. And if this GI is not here, then you're just gonna have a cable of length L, and your excitatory input is gonna, like the current is just gonna flow through, through the cable to point L. But then once you have this conductance here, now there's 2 paths for the current to flow. So this is not, like this EI is not generating any extra current, but this path is basically shunting current away from here. So the idea is this point is going to reach excitation, or somewhere further down is going to reach excitation if this is on and this is not on. Does that make sense? That's like the and not part. So the goal for this type of question would be to find the ratio of voltage when both of them are on, which we're going to call that VEI at L. So that's like your excitatory and your inhibitory input are present, over VE of L, which is when only your excitatory input is present. So that's basically like how much does the inhibitory input attenuate like your voltage at point L, which is like your goal point. So like how much are you shunting away from that goal point? So the strategy for this would be first to find VE of L, which is when you have only your excitatory input. And then the second would be to find when both are present, so VEI of L. And then you can just take the ratio. So for part 1, we're looking at GE alone, so we can ignore GI. So this just turns into a cable of length L with a resistor here with conductance GE and a voltage source, EE. And then this is point 0, this is point L. So first we need to find VE of 0. So like what is the voltage here? Because in order to find this, and using your, oh no, I erased the relationship, but the V of x over V0, in order to find what V of x is, you have to find what V of 0 is. So VE of 0 is equal to the current at point 0 over the input conductance at point 0. So what is the current at point 0? Current is just equal to voltage over resistance, which is voltage times conductance, since conductance is 1 over resistance. So that means that you know what this EE value is, but you don't know what this voltage value is. And so current is equal to the potential difference across this resistor times the conductance of that resistor. So that's EE minus VE of 0 times the conductance value, GE. Yeah. And then you can, we'll get back to that. N of 0 is, so this input conductance here. And then we're given that G out is equal to 0. So using the relationship in the equation before, we have that G in of 0 is equal to G infinity times tanh L, since this is length L. So then we can put this together and get that VE of 0 is equal to EE minus VE of 0 times GE over G infinity tanh L. And then we can solve for VE of 0. So that's just algebraic manipulation, and get that VE of 0 is equal to EE times GE over G infinity tanh L, oh gosh, plus GE. Okay, so now we have VE of 0. Now we want to find, let's see. Is it okay if I slide this up, or are you guys still, I'll like, okay. I'll also post these notes, so like if you don't get anything, you can look it up on Piazza or Blackboard. Oh my gosh. Okay. So, actually, it's short. So VE of L. So basically, we're just going to use the equation for V of X over V of 0 to find VE at L. And that basically simplifies to, so remember that our G out is 0 at the end. That was given in the problem. So that basically simplifies to hyperbolic cosine of 0 over hyperbolic cosine of L. Whoops, there should be VE of 0. Which is just 1 over hyperbolic cosine of L. So then you can use this equation for VE of 0 and plug it in, multiply that by 1 over hyperbolic cosine of L, and you get your VE of L. So that's the potential at L when you have only the excitatory input. So that's like just kind of similar to the one input problem that we had. So the next step is to look at when both the excitatory conductance and the inhibitory conductance are non-zero. So that's like when you have both the excitatory and the inhibitory input. And that's to find VEI at L. So now we're back to looking at this diagram. So the first thing is to find the G out value here. So G out at Li, which is just the G out for this branch. And then that is equal to this G in here plus this G in here. So that's like, again, just the branching law, or like the two resistors in parallel, essentially. So G out at Li is equal to G in, we can call this G in down, plus G in right. G in down is just basically the conductance that you see going into this branch. So that's just little gi, the inhibitory conductance. So this is gi. G in right is, again, we have to use that equation. So G in is equal to G infinity times G out over G infinity, etc. And so we are given that this G out is equal to 0. So this G in right is just G infinity hyperbolic tangent of the length of this branch, which is L minus Li. So then G out Li is just little gi plus G infinity hyperbolic tangent of L minus Li. Yes. Yes.
The big G's and the little G's, like, are they the same unit? Or are they? I thought like we would calculate in these notes, like we would calculate like the big G would be plug in something with the little G.
Okay. Okay. Somebody correct me if I'm wrong, but I think that for this problem, you can assume they're the same unit. It's just like this is the this is some conductance they give you, and then this is the big G's are also conductances. I think they should be the same unit. Yeah. Yeah, I think because there's the whole like membrane resistivity or conductivity thing versus like resistance or conductance where you have to do a conversion. But I think they would give this to you just in conductance. Yeah. Unless it explicitly says like the per unit like resistivity is, and then you would have to convert. Okay, so we have G out Li. Now we want to find G in 0. And then again, we're just going to use the formula. So we have the out conductance of this branch. We want to find the in conductance of this branch. So G in 0 is equal to G infinity times G out Li over G infinity plus hyperbolic tangent of Li over 1 plus G out over G infinity times hyperbolic tangent of Li. So just make sure the main things to note for this, like plugging in the formula, is just make sure you're using the right length and make sure you're always looking at like the G out that is of the branch that you're looking into. I think those are like the main possible things you could mess up. So now that we have G in 0, we can find VEI at point 0. So this is a little bit trickier. We need to use the fact that the current exiting through the resistor GE is the same as the current entering the rest of the circuit. So this is EE, GE. Here's the rest of the circuit. So this current, we're basically going to like make these two equal to each other. So I is equal to voltage times conductance. So we're going to look at the current exiting this part of the resistor, which is VEI at 0, which is here, minus EE, this is the potential difference across this resistor, times GE, is equal to VEI of 0 times G in 0. So this is the current entering the rest of the circuit here. And then you can solve for VEI of 0 and get EE times little GE over G in 0 plus GE. And then you would plug in your value for G in 0 from up here. So now we have VEI of 0, and then we can find VEI at point Li. And then that's just plugging in the formula. So that's VEI at point Li over VEI at point 0 is equal to, hyperbolic cosine of Li minus Li, which is hyperbolic cosine of 0, which is just 1, plus G out 0 over G infinity, or sorry, G out Li over G infinity, times hyperbolic sine of Li minus Li, which is 0, over hyperbolic cosine of Li plus G out Li over G infinity times hyperbolic sine, oh gosh, sorry, this is bad, of Li. So that's just plugging in the formula. And then you can plug in all of the values that you already have calculated. And then we can basically do the same thing again, since we have the input conductance and the output conductance, which is 0 for the branch L minus Li, and calculate VEI over L over VEI of Li. That's just, again, plugging into the formula with the input and output conductances you have. And then finally, once you have that, you can find the ratio VEI of L over VE of L, which is what we calculated from part 1, and that's when you have only the excitatory input. Yes?
Can you go through that again?
Sorry, which part?
Can you just like go through like what you've written again, since it's kind of hard to...
Oh, sorry, is this really bad? Okay. Where do you want me to start?
Basically like the VEI of Li divided by 0.
Oh, sorry. Okay, so
EI
basically what we've been doing up until now is like calculating the conductances, the input and output conductances for each of the like parts of the branch, right? And then you have this formula that relates voltage at point X to voltage at point 0. And then you're basically just plugging into that formula. So that's 1 plus G out at Li over G infinity times hyperbolic sine of 0 over hyperbolic cosine of the length of that branch, which is Li, plus G out of Li over G infinity times hyperbolic sine of Li. And I'll post these notes, so in case I can write that also here. Okay. Does anybody have any other questions on the and not gate and like how that works? Yeah.
In the non-use section, we calculated the input inhibitory input. Is that?
The input, oh yeah, that's another, so that's like another thing you could do. I don't think you need it to solve, if the question is like what is like the voltage at point L for when both the excitatory and the inhibitory input are present over the voltage at point L when only the excitatory input is present, like you wouldn't need to do that. But you can also look at when only the inhibitory input is present, and that would basically be like, you'd basically be ignoring this part of the circuit and just looking at like if you have a voltage, or if you have a conductance here and a voltage source, like what happens. Yeah. Would you have just inhibitory input? Let's see. So I think your G in value would just be GI, and you wouldn't have like a voltage source. Let me double check. Okay, sorry. So you remove. Yeah.
I know that. I spent like an hour doing that. So for VE, they just ignore VI, right? Then when he calculates VI, he assumes EI is not 0.
Oh,
and
okay.
to make math simple in this thing, he assumes EI is 0 because otherwise you need to have a contribution along the branch of EI. But because take that one and don't use like any voltage superposition. So for like when he calculates VE, EI is not 0, and then he's like assume shunting inhibition, and then for this part when it's like due to both of them, he assumes that's 0.
Okay. So yeah, actually, so I may have misspoke earlier. You wouldn't ignore this whole part. You would ignore like this part, and then you would have basically two possible branches.
So if it's providing voltage, how is that inhibitory?
Okay, so in that case, if you're looking, you would assume that so for the inhibitory like part of this entire problem, you assume that EI is 0, but then like you said, you could assume that this is some non-zero value, and it's just basically providing some voltage into the branches.
And in that case, it wouldn't be inhibitory if it was positive, if it was contributing positive.
Yeah, yeah. If it's contributing positive.
No, it can be inhibitory because it's negative. It doesn't make any assumptions on what the nature of the voltage is for that part. His derivation is honestly like quite bad. Like, it makes random assumptions in the middle, and like you really have to look closely in order to understand that he made them.
So yeah, if it were positive, it wouldn't necessarily be inhibitory. Does that make sense? But because the reason it's called inhibitory here is that this is not contributing any voltage, it's just contributing another path for the current to run down. Is that okay? Let's see. Okay, so in terms of Winslow, what I would worry about most is what he spent most time on and like what you have gotten the most problems on, which is essentially cable theory. But he does have 2 other lectures that are about like compartmental modeling and the dendritic spines. But the derivations for those are like quite complicated. So it's although it's good to know like how they work and the general concept behind them, I would think in terms of Winslow, like think more about which types of problems he could reasonably ask you on the exam versus like reproducing all the derivations. But so for dendritic spines, the main point with dendritic spines is that dendritic spines can change their length to change basically the degree to which a voltage change at the head of the spine is felt at the rest of the dendrite. So think of it as like, this is your dendrite, and you have a bunch of little protrusions. These are your dendritic spines, and you basically have a synapse here. So you're inputting some current here, and it's going to flow into the rest of the dendrite. And then the question is like, how does the voltage that is felt at the head of the spine, or like how does the voltage that is felt, at the dendrite, like here and going into the rest of the dendrite, relate to the voltage that is felt at the head of the spine? And so the main result from his lecture is that increasing length of the spine neck will lead to a decrease in the voltage felt at the dendrite compared to the voltage felt at the dendritic spine head. And the way he does that is by looking at this like simple circuit model of what a dendritic spine is. So this is some voltage source for the synapse. And then here's some resistance of the synapse. And then here's like a capacitor that stores charge. And that's like the dendritic spine head. He also has a resistor that represents the dendritic spine head membrane. But the assumption that he makes is that this resistance is huge since the membrane is like tiny surface area. So you can just ignore it because there will be essentially no current flowing through it. And then this is the voltage felt at the head. You have some resistance associated with the neck. And then here's like the rest of the dendrite. And this is the voltage felt at the rest of the dendrite. And basically your goal for this type of thing would be to calculate VD over VH given these values. So let's see, how are we on time?
We still have a couple of sections.
Okay, yeah, so this derivation is like pretty good in his lecture. And I would say the main thing to remember is that as you increase the length of the neck, you decrease VD over VH. And then the other thing to remember is some of the assumptions he makes. So there's no current through the head of the membrane. There's a really short neck, so you can ignore any cable properties. Just treat this as a single resistor. And then you can ignore any like membrane resistance for the neck. So you can basically, there's no input resistance going into this part. So it's just a very simplified circuit model where you ignore all the cable properties. And you just have this circuit. And then the very last part of Winslow, which was lecture 2, is the compartmental models. And again, like he goes through the whole derivation in his lecture. And you can basically like have a bunch of compartments, make matrices, get like a steady state response. I would be just like familiar with how the equations are set up, but don't worry too much about the details of all the derivations. But the main kind of result from that part of his lecture is that if you assume a conductance change at like where you're inputting current as opposed to an actual input of current, you get a nonlinear relationship. So if you keep increasing the current, like if you keep increasing the current that you input when you change the conductance, you're not going to just keep getting an increase in resulting voltage. It's going to be like that sigmoidal relationship that he showed. But again, these are more like I guess, conceptual, theoretical things that he showed. So that is it for Winslow. I will post the rest of these notes, which have a little bit more detail. And then all the details of the derivation are what Winslow gave. Sorry, I had to rush through the last part, but I want to leave some time for the other. How does the do you know how the projector goes down? Oh,
Do you need the board? Do you
Yeah, I
need
need
slides
the
or board?
board.
Board.
You don't use slides?
Is the audio showing up on it? You can see the, yeah, cool. Okay, so I'm going to go over the stuff that Dr. Zhang and Dr. Vogelstein covered. They only had one lecture each, and neither of the lectures were particularly material heavy. So this will be pretty quick. Definitely, focus more on the first couple professors that we covered because they had the majority of the lectures. But I'll quickly go over sort of what was covered by these two professors. Are there any questions before I start? Yeah. So, yes.
Every professor that you've had so far has a section.
Right. Yeah. It's in proportion to how many lectures they had. So, yeah, Vogelstein will be on the exam, but it'll be a very, very small part of it. Same with Dr. Zhang. The vast majority of the points will be Winslow and Dr. Wang. So focus on those. But yeah, the other professors are technically on it. Any other questions? Again, I wouldn't worry as much about these professors. Worry more about the ones that had more lectures, okay? All right. Any other questions before we get going? Can everybody hear me? Yes. Yeah? Okay. All right. So, Dr. Zhang, or Zhang, however you pronounce it. The first thing he had was the delay line model. Actually, Dr. Winslow also talked about this. So, yeah.
Are you going to be tested on song stuff as well as special?
Yes. Yeah. Yep. But remember, if they only had one lecture, they're only going to be a very small part of the exam. Okay? So, don't spend half your time studying, one professor who had one lecture, even if they had a lot of material on it, in that lecture, okay? What?
We're not at liberty to speak on that.
Oh, they totally have.
Have I seen the exam? Are you asking if I've seen it, or? I've seen it. Yeah. I can't tell you what's on it, but I've seen it. Yeah, of course I've seen it. I'm a TA. All right, well, the whole idea here is, especially with the auditory system, you have an input coming in from the left ear, an input coming in from the right ear, and then there are neurons that are connecting to more neurons. These are sort of those connections. And these are sort of like AND gates, and they each go off on another neuron. So what you can see here is that if the left input comes in before the right input, then it'll travel along further before this one comes in, and they'll both hit this one at the same time. And since these are AND gates, the output neuron will only fire if both of the input comes in at the same time. So this is kind of a model that it can encode sort of the time delay between the two ears, and then therefore you can infer information about, maybe where that sound came from, right? Any questions? Cool. Next we have a perceptron. All right. So with this, the idea is that we're computationally modeling a neuron. You'll have several inputs. We'll just draw a few for now. Each of these inputs will denote X, X1, X2, X3. Each of these is sort of like a neuron that's coming in to the system. And each of those is weighted. So you're weighting your inputs. That can be thought of as the strength of the connection at the synapse. And we will learn more about this when he's going to lecture more for the second test. So we'll probably get back to this a lot. But then you take all those inputs and you sum them. All right. And then there's also a bias term. So with a bias term. So there will be some sort of offset, some scalar offset to that. All right. Which we can denote H. And then this, are you still getting audio? It's a little, yeah? Good, okay. Then this will go through some nonlinearity function, usually a sigmoidal. All right, and then an output. So we have these inputs. They'll usually also have gone through that nonlinearity, so those will all sort of be 0 to 1 in this model. We weight those, we sum them, we add a bias, and then we do a nonlinearity, and then we'll have either, well, the value 0 to 1, probably it'll be closer to 0 or closer to 1, and that'll indicate whether this perceptron or neuron model is going to fire based on the inputs. All right? So we can write this out mathematically as y equals g of the summation over I of w times x minus h, where x is our inputs, w is our weights. We sum those all together. We take into account some scalar bias. And then g is our nonlinearity function. All right? So does everybody kind of see how that equation lines up with that drawing? Yeah? Any questions? Yeah?
There's not a bias associated with each loop, right? It's just one bias?
Right, there's just one bias. And the idea there is that when there's no input into the system, and all those are 0, there may still be some sort of firing there, or some sort of output. So that's sort of what the bias is doing. Yeah?
Is that H within the sum?
No.
Okay.
No. Yeah, outside of the sum. Yeah. Yeah?
Is where in the process is G applied? I'm sorry if you already said it, but is it applied like on X, or is it somewhere else after that?
So G is this block here. So it's sort of applied to the whole, weighting of the inputs plus a bias. Yeah, on that whole thing we do G. Yeah? Yeah, I'm subtracting, but you can also add. It's a scalar H. It could be positive or negative. So it doesn't really matter if you say plus or minus. All right. All right, so we can construct a network using a bunch of these things, and we can change how we're formatting things a little bit. So we're going to talk about neural nets. So you'll have output nodes. This may be y1, y2, y3, y4. And then you'll have some input nodes, x1, x2, x3, x4, x5. And then we have our weighting. So for each of these connections, we have the summed weighted input from all of these. And we do that for each one of our outputs. All right. So each of those lines denotes an input line where the input is weighted by some factor and added to what the output will be. So if we were to write this out, it would simply be yi equals the summation over j, wij, xj. Notice in this very simple model of a neural net, we're not doing that nonlinearity, and we're not really doing a bias here. We're simply weighting the inputs and summing those to the output. Yeah?
Each of the lines from x1, for example, are they the same, or are they each weighted separately?
They're each weighted separately. Yes. Now, if you wanted to take into account a bias, you could add an extra dot here and call it h, and have all those weights going. And then if you wanted a linearity, you could put a box in the next layer that weights those, but we're not going to do all that right now. Just very simply, yi equals a summation over j of wij xj. So we could write this. This is for every single output, right? For every I. For all I, where I could equal 1, 2, 3, or 4. But we can write this out in a matrix form to solve for it all at once, where y equals w x, where w is a matrix of wij elements, x is a matrix of xj elements, and y is, or it's a vector, and y is a vector of yi elements. And doing this, you could solve the whole thing with one quick matrix transformation. Yeah?
Would every single line have a bias, or would it be like every output is a bias?
So if you wanted to add a bias, what you would really do is you would treat the bias as just one additional input. And then you would connect all those lines to the outputs. Therefore, each output would have a bias with a weight. And that weight would sort of determine the bias for that output. Yeah?
I'm really sorry, but the major distinction between the neural net and the perceptron is not the multiple outputs, it's the fact that there are more than one layer.
Sure. Is that the actual definition, or are you asking?
That's more of a definition.
OK, so there you go. We could call this something else then if you'd like. Maybe multi-output perceptron, right? That's simply what he had in the PowerPoints, I think. But for those who want to stick to the exact definitions, multi-output perceptron. Any other questions? Yeah?
Would you test on anything more complicated than like hidden layers and what connected?
You will not be tested on anything more complicated than what was covered in the lecture. Any other questions? OK. So there was an example that went along with this. Does everybody have this? All right, so using our multi-output perceptron, or even a full neural net with multiple layers, we can look at an example with auto-associative memory. So the idea here that he presented in class was that you present a face to a neural net, you train it, Maybe you present a bunch of pictures of the same face to the net or the multi-layer perceptron, and then the weights will be adjusted. That process can be done in a number of ways that you don't really need to worry about too much right now. But you want the output to be the same as the input. So if we have input x, what you want is output x. All right? And then the idea is, if you have the image x and maybe half of the pixels are dead or aren't there, you still want to reconstruct x based off of your training. So we have x equals wx. That's the goal. And the weights after you train can be x times the pseudo-inverse of x. All right? And the pseudo-inverse, we're saying that's not a true inverse simply because there's no guarantee x is a square matrix, right? But that will be your weights. And using those weights, you can reconstruct your output even if a lot of the input is gone. And that's called auto-associative memory. Any other questions about that? Yeah. So that would mean that basically in our equation y equals wx, where these are vectors, right? Or they could be matrices, but let's just say vectors where we're kind of getting the picture into one long vector. We're just saying that y equals x. The output is going to be whatever the input is. You're just reconstructing it. Yeah.
Why is W not just an identity?
Sure. So it can be, but if it's simply an identity matrix, then when your input no longer has all of the pixels or some of them go dead, then you're not going to be able to reconstruct what the full input is if it's just the identity matrix. But if you have it as x times the pseudo-inverse of x, then you can.
So is x like the whole image, or is x like part of the image?
X would be the whole image. Well, which x do you mean? Do you mean the original x that you're training on? Yeah, would be the whole image. But you may say, all right, if we input a vector, maybe it's not x, but let's call it z, then you still want to get x, where z may be x where several elements have gone to 0 instead of whatever they used to be. All right? Yeah? That's a good question. So it's a vector. It may be that you have a picture where the picture is a matrix, where each element is a pixel in that matrix. But what you do is you simply concatenate all the columns so that you have one extra long column of all the pixels. Yeah. Okay. So the only other thing that he really talked about was orientation / position mapping. So in the brain, how do we keep track of orientations and positions of ourselves in the world? A lot of these experiments are done on rats, but a lot of it can still be applicable to humans. First is the hippocampal place field. All right, so for this, firing rate of neurons are a function of the position due to landmarks. So you may enter a room and note that, there's a table over there. Then, you have this place field in your brain where neurons will fire depending on where you are to that place field, or to that landmark. There's head direction cells. All right, so these are not these cells don't fire as a function of where you are compared to a landmark, but they fire compared to what direction you're facing. All right, and with that, that's mainly calculated due to there's some orientation that we'll just say is the zero orientation. And then your body can keep track of how you're moving, what sort of angular velocity you're rotating with, and it can then calculate which direction you're facing after a movement. And then those can be corrected using landmarks that you can see. So maybe you turn and your nervous system thinks, okay, I'm really 50 degrees this way. But then, I might see the monitor and be like, oh, I can't be exactly 50, I must be 45. So it's kind of suspected that that may be a prediction correction model that's going on there. And then there's grid cells. So these are sort of periodic spatial locations, and there are several different scales. So one group of grid cells may be where you are on campus. Another group of grid cells may represent where you are in a building, and so on. So those are sort of different resolutions of position. And I think that's really the only three he introduced in his one lecture. So yeah, any questions?
Could you repeat the first one just really briefly?
Yeah, hippocampal place fields. So that's your position relative to landmarks. Yeah.
Will we be testing on more conceptual stuff, or is it going to give us a question we have to do something?
I don't think I can really say. Both are fair game. Yeah.
So the Zane practice questions that are posted online, are those like even relevant to which like should
What were the practice I didn't look at them. What are they?
I think you're referring to the past problems that have been posted. So that's so last year, Zane had more lectures for exam 1.
Yeah, he had a few.
Yeah, and so those practice problems are based on material that's a little bit more developed than the material that he went over with you guys. So your guys is much more introductory. So I would say don't necessarily depend on those practice problems to the level of you only need to go as far as
I would suspect that it is also using landmarks, and yeah, I would suspect a lot of this uses landmarks as well as information that your brain gets about how your body's moving. So if you were blindfolded and you move 10 steps in a direction, those grid fires would attempt to fire saying, okay, on this scale I moved 10 feet in that direction. But I'd imagine that that's also corrected with landmarks. So does that answer your question? Yeah, I mean, he didn't cover too much about how all these things are calculated, so I can really only guess at it. Other questions? Okay, so Vogelstein had a really laid-back lecture. He kind of just talked about what is data science, what is biomedical data science, perhaps what are some good systems to use. So I can go over those briefly. So one question he asked is, why is it hard? Why is biomedical data science hard? So the first answer was volume. In biomedical data science, there's usually a lot of data, right? There's a large volume of data, and that in of itself can be a problem. There's a large variety of data, all right? It depends heavily on the patient. It can depend heavily on the day the data is collected. There's a lot of variety in the data. And actually, that may be a better description for veracity, but we'll get there. Velocity. Most of the data is streamed, so you have to take into account considerations of how fast is that data streamed, what's the sampling rate, how quickly can you acquire the data. Veracity, a lot of the data is very noisy, and so you need to think about how are we going to filter that data, what kind of noise is being introduced into the data by various electrical and mechanical systems. As I'm sure a lot of you know, when you collect a lot of medical data, there's usually 60 hertz noise due to the medical equipment that's used to collect that data. It's just an example, but a lot of that will have to be filtered out. And of course, domain knowledge. It's not just the data itself that's complicated, but medicine itself and biology itself, there's a lot of domain knowledge that you might need to have to even begin thinking about analyzing the data that you're going to collect. So those were 5 points that he wanted to make clear. Why is it, difficult to do biomedical data science? And then he also asked what kind of system tools are required. What kind of tools do you need to do biomedical data science? You have to have some way of managing the data. This includes databases and stuff like that. Storage, how are you keeping track of all this data? You have to be able to visualize it, which is not always easy. A lot of times this data may be high dimensional. You need some way to accurately display the data that you're managing. Wrangle. So this are processes that you're doing to the data that doesn't really change the data that you're representing itself, but perhaps it may be things like outlier detection, convolutions, stuff like that preserves the data's identity, but maybe you're cleaning it up or representing it in a slightly different way. Summarize. So you have to have some way of summarizing the data that you have. You can't just show somebody a table with 6-dimensional data. You need to be able to concisely express what that's showing. So that's stuff like principal component analysis, just taking the mean, confidence intervals, stuff like that. Generalize. So that's stuff like measurement bias and estimators. Predict. So perhaps using the data to predict things. That's classification, regression, some things like that. And then control. Using the data to control a model, perhaps things like Kalman filtering or reinforcement learning and stuff like that. So those are, what is that? That's 1, 2, 3, 7 points that he kind of introduced to you guys about what kind of tools you're going to need, what kind of systems you're going to need to tackle biomedical data science. Any questions? Okay, well that was all there really was to his material. So that's it for them. Who's up next?
There we go.
Yeah, in the old version. All right, so these 2 sessions are pretty easy. I would say at least you don't need to pay much attention to it. Just to go over the big pictures, and I think we should be fine for the exam. So Dr. Cullen talks about the sensory motor system. It's just the introduction. I think she will talk more in the future, more like difficulty ones, but here is pretty just the introduction of your central nervous system. And Dr. Song talks about the optical imaging. I know the concept is a little bit hard, but just the big pictures here. You know, how does that, I mean, what does each kind of optical imaging works, and then that should be fine. So first, the sensory motor system, or the basic concept of the CNS. Central nervous system, we need to know the development of it, some simple anatomy or the structure of it, the building blocks, what the functions of each building block, just the very basic ones. And then you need to know the microstructures of the neocortex. It basically just the 6 layers, and some general signal pathways for the sensory processing and motor processing. So what is CNS? CNS is just the central nervous system. We have the cerebrum, which is the cerebral cortex, and also the cerebellum and the spinal cord. So we also have the brainstem here. So if you cut it sagittally, you will see there is this big chunk of cerebral cortex. There will be a diencephalon, and then it is connected to the whole brainstem. The brainstem composed of 3 parts: the midbrain, pons, and medulla. And also there is a cerebellum after the brainstem, and then it is connected to the spinal cord. So for the development of CNS, you need to know that how our brain or how the central nervous system actually formed from a baby to a normal adult. So at first, our central nervous system only composed of 3 parts: the prosencephalon or the forebrain, mesencephalon or the midbrain, and then the rhomencephalon or the hindbrain. So this is the first stage, only have 3 vesicles. And then it will be developed into a 5 vesicle stage, in which the forebrain or the prosencephalon has been developed into 3 different parts. We have the telencephalon and the diencephalon, and also we have the optical vesicles. So the optic vesicles just will develop into the retina. The telencephalon here is more important. It will develop into the cerebral cortex, the biggest part in our brain. And the diencephalon here will develop into thalamus, hypothalamus, and yeah. So, and we also have a midbrain here, which is not actually developed much compared with the size here for the prosencephalon. We also have a hindbrain, which will finally develop into the other 2 parts of the brainstem, including medulla and pons. So this is the second stage, the 5 vesicle stage. Then, yeah. Oh, here, yeah.
I have a question. Since there's a lot of like anatomy,
yes,
like in general for
yeah,
this section,
yeah,
what can we be expected to know for like each of this? Are we kind of like have the label or like the functions or?
I would say have some general memorize the general anatomy, like the big part, the big lobes, and different, like the development. You need to know there are 3 vesicles first, like each the name of 3 vesicles, and then for the 5 vesicle stage, and then for the whole brain stage. But just the big chunk, you don't need to know details.
And probably you need to know how does that look like. If you want to draw it, how do you label it? So I would say yes, the anatomy is important here, but not the details.
Is there a general outline what differentiates general and
that line is hard to draw.
details like
That I really cannot say, but
Use the lecture recording and use the lecture slides. That's
yeah.
that's all of it.
All right. Okay, so let's continue then. So when the CNS is at a premature stage, it actually it looks like a tube. So in those between those tissue, there will be this kind of holes, or there is a tube. So this kind of stuff here, the structure here in between, will be composed or will be filled with the CSF, the cerebrospinal fluid. So it will finally develop into the ventricular system of the brain. So as you see here, the two lateral parts of the telencephalon will be developed much and into the cerebral cortex final. But the other part, the diencephalon, the midbrain, it will just generally not develop that much. So this is a mature brain that we have several parts. These parts are first defined by those anatomy divisions, like the central circus, this kind of lines. But different lobes are different in their functions. So we have the frontal lobe, which is more for the motor functions, executive functions. Parietal lobe is more for the sensory informations. Occipital lobe is more for the visual functions. Temporal lobe is for auditory and some memory and more like associative functions. And we also have the cerebellum here is for the coordination and movement. For the brainstem, it's more like connect your cerebral cortex with the spinal cord, and it is important in the sensory and motor pathways. If you cut your brain like in the mid-sagittal plane, you will see there is a corpus callosum, just the white matter that connected the two hemispheres. And you will also see the ventricular system, which I think I have it, the second slide. So this is the ventricular system of your brain. So as I said, in the ventricles, you will have the CSF, like the fluid. It will help to, it sort of baths your brain inside of these fluid conditions. So it helps to protect the brain. So for the ventricular system, you have two lateral ventricles, and then it will go into the third ventricles here. And then after the cerebral aqueduct, it will go to the fourth ventricle, which is in the brainstem, and finally go to the spinal cord. So when I took the medical class, the teacher said that the structure of these ventricles just look like the spaceship in the Star Trek. So I don't know if it helps, but it just helps you to memorize the shape of it. So yeah. I would say remember the lateral ventricle, third ventricle, fourth ventricle, cerebral aqueduct. You know the name and you know how does it look like. Yeah, that's pretty much it. So this is a comparison of your human brain and the rat brain. We know that the humans are so unique because their brain is large. So we have several differences if you just compare the rat brain and the human brain. So first, there are a lot of convolutions in the human brain. You have the grooves and bumps. The grooves we call it sulci, and the bumps we call it gyri. We also have the organization of the human brains, especially for the frontal part. As I said before, the frontal lobe is more for the motor functions and executive functions. all your personal feelings, the personalities are like more controlled in the frontal part. However, for the mouse, we know that it has a pretty sensitive, I would say the, let me see. So the olfactory bulb, yeah, it should be the olfactory bulb for the rat is pretty large compared with humans. So it is more for the smell functions of the rat because rat is pretty sensitive in smelling, but not our humans. We also have the brainstem orientation. It's different for the rat and for the humans. As you can see, for the rat, it is easy to define what is rostral and what is caudal. Like in the anterior part, it's just called rostral, but we also have the caudal direction. So the brainstem is just this horizontal or the anterior to posterior direction. However, for the human brain, we have a turning point of the central system. As you know that our spinal cord is from top to head, from head to foot. So it's like from superior to inferior, but it has a turning point at the brainstem. So when you actually go to the cerebral cortex, it is from anterior to posterior. So it's like this way. It just does some basic orientation of the central nervous system. It helps you to define which part is lateral or medial to the other part or anterior or posterior. I think she also talks about the microstructure of the neocortex. So the neocortex is not just a simple cortex. It has several structures of the neurons or the cells. It has 6 layers. The 6 layers are different in terms of the shape of the cell and the functions. For example, the 4th layer is more responsible for the input of the signal, while the 5th layer is more for the output of the signal. So, for example, if you look at the primary sensory cortex, because it is more like sensory information input, it will have a very rich layer of layer 4. While if you look at the primary motor cortex, it is more for the function, the motor function, so it has more output of the signal. So for that cortex, you will see a very rich layer 5. Yeah, so this is just the example I talked about before. As you can see here, the primary visual cortex is also have a very rich layer 4, but if you look at the motor cortex, it has a very rich layer 5. So she also talks about the general signal pathways for the sensory and motor processing. I think the basic principle for both sensory and motor is that your right side of the brain controls the left side of your body, and the left side of the brain controls the right side of the body. So it has a cross point for both sensory and motor functions. For the sensory processing, when you touch something, for example here, and then the signal will go into the spinal cord, cross at the spinal cord midlines, and be sending into the brainstems. After the brainstem, it will go to the thalamus for the third level processing, and then finally it will go to the primary sensory cortex. And here the processing will be taken, and then the information will be passed down to the other associative cortex and to give other instructions. So it has several layers. First the spinal cord, then the brainstem, and then the cortex. It's similarly for the motor processing. The motor processing is from your brain to the spinal cord and to the effectors, or for example your hand and your feet. So first it will go from the motor initiative will be at the primary motor cortex, and then it will go to the brainstem. After the brainstem, it will cross the midline and somewhere called pyramid, and then it will go to the spinal cord. At the frontal horn or the ventral horn of the spinal cord, it has a large bunch of motors, alpha neurons, and then those neurons will fire the action potentials and instruct your hand to perform certain certain movement. Yeah, so primary, so this slide just tells you that not only our motor functions help to initiate the movement, why your movement is so coordinated, it is it depends on not only the primary motor cortex, it also depends on the thalamus and also the cerebellum, both and the basal ganglia. All these structures helps to coordinate and to adjust your movement. Yeah, so I think that's it for the Coulom's lecture. So just a big picture here. And then Dr. Song talks about the optical imaging. I think for optical imaging, I know the basic principle for each technique is pretty hard. I don't know that very much in detail, but I would say the most important thing here is to understand how to use this image to image what you want. Like the difference between each technique. For example, I give you something and you can choose which one you want, the application of this optical imaging technique. So he talks about the fluorescent microscopy, which include the confocal and the two photon and the light sheet. He also talks about the intrinsic signal imaging and the calcium imaging. So first, why our brain is so hard to use the optic imaging. So we know that when the light goes into the brain, it will be absorbed by the tissues very quickly, so it will decay very quickly. It is really hard to go through the brain. It's not transparent, so you cannot see the deep part of your brain with optical imaging. And also the light will get scattered when it goes into the tissue. So we have different kind of technique to help the microscopy to essentially we want to image more the deeper tissue of the brain, not just the surface. So the first technique he talked about is the confocal fluorescent microscopy. So the confocal is essentially when you have a light source here, you use a pinhole to restrict your light into a very small point, and then when you focus it with the condenser here, it will be focused only to one point on your tissue sample. So it is easier for you to get a very high resolution of image. So you just use the pinhole to focus your light into each point of your samples, then you will be able to get a very high resolution image. And also with this focusing of the light, it will decrease the scattering of your the light and decrease the decay of the light. So like for example here you have a wide field fluorescence and a confocal fluorescence. The two image shows that the confocal has a pretty high resolution. However, the other one he talked about is the two photon image. The two photon means that instead of using one photon to excite the fluorescent in your tissue sample, you can use two photon with a shorter wavelength. So with this two photon, the advantage of the two photon similarly is that the focus or the excitation can only be achieved when the two photon focal point are met. So like each photon will have a cylindrical, each photon have a focal point, and the resting will be like have a low energy. But when the two photon excitation can only be achieved when the two photons focal point are met. So it will help to increase the image resolution as well. So for example, here, as you can see, if you only use one photon to excite, it will have a lot of scattered photons. And yeah, so the resolution will be very less, and also you cannot image deeper tissues in the brain. However, if you have a 2-photon microscopy here, the focus will only be met here. So you can only image this single point in your samples or in vivo, and it will help to increase the resolution and increase the deep depth of your image. However, there is a problem for the 2-photon image, which is the photo bleaching and the photo damage. It is just a status that when your photon reach a triplet status, it cannot come back. So it is a general decay of the signal or the excitation. Similarly, as 2-photon, you can also do a 3-photon microscopy. You use a even less or even shorter wavelength of photons to excite the fluorescence in your samples, like this one. And it, yeah. Sure.
So what is the advantage of 2-photon, 3-photon scanning versus confocal?
I would say confocal is a 2-photon, and the 3-photon will be can help you to image deeper, and also can achieve even higher resolution compared with confocal. And the scattering of the 2-photon is less compared with confocal. Yeah.
So like, why would you ever use 2-photon instead of 3-photon? Just like for cost, or?
You mean like why we use why we not just use 3-photon? Yeah. I think that's probably because of the hardware. I don't actually know, but 2-photon is more widely used nowadays in research. It's probably because of the hardware, which is hard to generate the 3-photon waves. Yeah, that I'm actually not sure, but yeah, not that detailed. The third one he's talking about is the light sheet microscopy. So the light sheet, instead of, as we said before, the confocal and the 2-photon is to image only one point, with a high resolution, one point on the tissue sample. But what if we know that if we want to image the whole tissue sample, it will take a very long time. You have to do point-by-point imaging. However, the light sheet is a method to help you to image the whole slice one time. So the light is to be restricted into a very thin slice here, so you will be able to get signal from only one slice in the tissue. Yeah, it is very fast, but it has a low resolution. So this is just a comparison of the three technique. As you can see, confocal use a pinhole, but it uses only one photon to to focus, and only one photon to excite your tissue samples. 2-photon use 2-photon. Light sheet is a a thin slice of the light to excite not just one point, but the whole slice of the tissue sample. So for the light sheet, it will be very fast compared with the confocal and 2-photon. But the photo bleaching, which we said is the decay of your light or sort of a waste of your energy, for the confocal is higher than the 2-photons. These three are all fluorescent microscopy. He also talks about some other in vivo technique. The first one is the voltage sensitive dye imaging. Essentially, this is just to inject a dye into your tissue, and it is sensitive to the voltage because of its chemical substances. And then when there is a action potential fired or the tissue is excited, and then you will be able to see the dye will emit some different wavelengths of light, and then you will be detected just using normal microscopy. The other one is intrinsic signal imaging. This utilizes the property that in our brain we have a lot of oxygenated hemoglobin and deoxygenated hemoglobin. It is just carried by the blood flow. And when your neuron is excited, it will use energy, it will use oxygen. So the blood flow will carry all this oxygenated hemoglobin into your brain. And the oxygenated and deoxygenated hemoglobin will have different sensitivity to different wavelengths of the light. So you can imagine that for a excited tissue, it will have a different absorption rate compared with the other part of the brain because the different concentration of oxygenated and deoxygenated hemoglobin. So use that you can just image to see where your brain is excited. So all these techniques are like in vivo. It can show the dynamic changes of your brain. And when, for example, when you give someone a task, probably you will see the firing of the tissue there. The last one he's talking about is the calcium image. Calcium image is pretty popular these days. It's based on the principle that when a neuron fires a action potential, it needs the calcium to flow into the neuron. There will be calcium channel open. The calcium will inflow into the cell and cause the depolarization of the cell membrane and the increase of the potential to firing a action potential. So the calcium image uses a calcium dye or a calcium indicator to bind in those calciums, and when it goes into the cell, it will be able to show it will be able to show where your cell is firing, where the excited neuron is. So essentially, when you inject a calcium dye into your tissue, into a living mouse, and then the dye will bind to those calciums, and when it goes into a cell, you will be able to detect that with the detectors. So this is just the image of it. I think that the optical imaging, just to know the application of it, I think that will be fine.
My laptop runs out of battery fast, so like if you or something, just let me know. Here. Is it on Google Drive?
It's on the Slack. Like I sent it in the thing. I'm not gonna I'm just gonna go fast because
I know.
They're like super antsy. Yeah. Is this it?
Oh shit.
Okay. Will it run out in like 15 minutes?
No,
or
no.
30 minutes?
It'll
okay.
stay.
I'll plan to go that long, so.
Okay guys, hang in there. Final professor.
Okay, perfect, thank you. Okay, so I'm gonna briefly cover Connor's part. So since you guys have been here for I don't even know how many hours, I'm gonna fly through it. So the idea is that I condense both of his PowerPoints into one single PowerPoint. All the bolded information in the notes part is included in these slides. So hypothetically, if I didn't screw up, everything's in here, and you should really be looking at the titles of my slides because that's where all the important information is. Okay, so just a quick run through. This is basically outlining the first slide that Connor goes through, and that's saying that unusable image information, like that you see in the world that first falls upon your retina, is distributed, implicit, and unstable, meaning that it's something that you can't process and put into neural codes. And so what your entire visual system does is makes it a usable representation of information we need about the world, which is then described as compressed, explicit, and stable. And so what he really stresses is that all of these themes throughout fall into each aspect of the visual system that he talks about, and that's what's important. So you should know when he talks about the V1 or the V4, what is that doing? Is that compressing? Is that making something more explicit? So my headers will have those words in red, so make sure you pay attention to those. Okay, so this is exactly what Connor put on his slide. I put a couple other red annotations just to clarify a few things that I thought were confusing, but that just clarifies the slide that I just went through. And then he shows the diagram on the right, which I thought was a little bit confusing because it seems like, okay, the light's going opposite where you think it would because it first gets processed by the rods and cones. But if you look at the diagram on the left, you can see how the light falls, and it actually does fall in the ganglion cells first, but it's processed by the rods and cones, and the neural signal is sent to the bipolar and then to the ganglion. And in terms of anatomy and stuff like that, you really don't need to know that much, right? Because he said that explicitly. So definitely what I would focus on is whatever I put in bold, because those are the things that he thinks are the main details. So if you look at what he talks about with the anatomy, he just wants you to know, okay, what do rod cells do? What do cone cells do? And you just need to know that rods are sensitive to light. So when you're in a dark room, that's those are the cells you're looking for. And then when you're looking at cones, they have different opsins, and so they're sensitive to color. And this will all be posted on Blackboard, so feel free to read through it. If you have questions, feel free to ask. So with rods and cones, the important part is how they're distributed and the different opsins or pigments that are found in the cones. So how they're distributed is in your eye, in each eye you have a fovea, and that part is where all of your cones are concentrated. And so there you'll have a lot of good detailed information, but you won't really be able to see darkness, which is why he mentions in his lecture that if you were to actually look from the sides or the peripheries of your eye, when it's dark outside, you would be able to see better. And that's 100% due to the distribution of rods and cones. So cones in your fovea right in the center, rods everywhere else. And then on the right side, I just put a quick diagram. You have 3 different types of cones. Each one has a different type of opsin. So these pigments have medium, short, and long. Sorry, I don't know why I said it in that order. Wavelengths. And so basically that covers your entire spectrum. Your relative activation of the different cones is the basis of your color vision. Great. So moving on to retinal ganglion cells. So now I'm moving away from the cones and rods and moving a little bit deeper. And so you start sending the signal from your rods and cones to the retinal ganglion cells. And these cells are pretty important, and this starts to touch on what you've learned in other lectures about receptive fields. But the cool part about these is that with your rods and cones, you have one spot that's your receptive field. But what the retinal ganglion cells have is what they call a center surround. So he does really stress this because it's super important with the purpose of what your retinal ganglion cells end up doing for image processing. So you have on-center ones, which are shown on the left. You have off-center ones, which are shown in the middle. And what this means is that in the center, based on if there's an increase in light or there's a decrease in light, you'll have firing or no firing. So I'll go through this a little bit slower here. So I put it all here. This is what you need to know. Basically, with your on-center ganglion cells, let's say right in the center portion, you increase light. So your on-center ones are going to fire. So you see all those spikes, right? Now your off-center ones, if you make it darker in the center of their center surround, now you see that you increase spikes then. So that's kind of the basis of why it's called on-center and off-center. But what else is super important is looking at the diagram on the right. So each of these has a surround section. So following whether it gets lighter or darker, this surround section will actually suppress what happens in the center. And that gets to be really important when I get over here, but we'll talk about that a little bit later. Before I move on, so basically the basis of having an on-center or off-center is because they synapse onto different bipolar cells. And so you have on-center bipolar cells and off-center bipolar cells as well. And I put a note here, this diagram's super misleading because you don't have two neurons synapsing onto one. It's usually just like one, like in the right diagram, but they just show this so that you know that there's off-center and on-center ones. And then super brief, you don't need to know the details of how this happens, but they're included in the notes in his slide, is the basis of having the surround suppress your center. Like basically like what happens in the center is the fact that you have horizontal and amacrine cells surrounding it. And so with like GABA and different neurotransmitters, you actually end up having that suppressive or inhibitive kind of function. All right. So I put this all here as notes just so if you get a little confused, make sure you come back to this and double check your knowledge. But this is what is more important. So you see how I put in the title like compress in red? Like he's like really into these like buzzwords, which are also really important because that's like how you get your signal from like when it first falls upon your retina to like when it's a usable image in your mind. So your first step is the retinal ganglion cells actually compress image information about brightness and color regions. So this was when he was talking about edge detection in class, if you guys remember that. Basically because you have the center surround effect and that kind of system, you're able to get a high response rate when you're detecting an edge. Because when it's completely in darkness, your on-center ganglion cells aren't going to fire at all. So they're not activated. When you're completely in bright, the center is activated, but also the surround is also activated. So now you have the center going and firing, but the surround is actually suppressing that. So you're getting a very low response rate. So what's important is when you see it's like crossing that border and you have an imbalance between your center and surround, that's what's important because now you're saying your retinal ganglion cells are compressing this information so it's more usable and you're detecting edges. Because it'd be really, really inefficient for you to be like, oh, I have like 1 million of these cells and they're just like detecting a white wall, which is what he includes in his notes. What's really cool about this is you're compressing because you're able to detect the differences.
So in detecting differences,
yeah.
like only some of the neurons would fire in that portion of the center surround ganglion?
Yeah, and then so you have like the difference between your on-center and your off-center. So depending on like what types of edges and like brightnesses you're like trying to detect. I don't know if that answers your question. Yeah, so you have like multiple. So a little bit, yes.
For E on the last slide, is it that there's no response? Like on the graph it looks like there's like a smaller response, but you wrote that there's no response, so.
Yeah, so it's like a low level of response that depends on, I don't know if this, because I don't have like the source of this diagram, I'm not 100% sure if it's like completely in the light, but basically the idea is that you would get enough suppressive activity that it wouldn't be like edge detection anymore.
Okay.
Does that make sense?
If you relate this back to the slide earlier, there is some amount of spontaneous firing when you have sort of a white center and white surround, or the on-center, off-center ganglion cell. So on-center, off-surround ganglion. So as you can see in sort of the red box, the third slide there that they're shown, there is some amount of firing. So it's not completely.
Okay, all right, so going back a little bit to the anatomy, again, I would say you like need to know general like big concepts, but you're not gonna have to like super memorize everything because like Connor just has never done that and he says that in his lecture. So what's important is that the optic nerves leave your eye and that's what's carrying that information. It goes to the optic chiasm, I think that's how you pronounce it, but basically that's the idea that left projects to right, right projects to left. And then the next area is the LGN, and so from the LGN then you go to the primary visual cortex, which is the V1. And he includes that little like colorful diagram because just like we were talking with the ear, there is like a map, so there's like a retinotopic map to the brain, which is kind of cool. All right, so the next important part is that your V1 actually compresses the information from a line of retinal ganglion cells responding to a local edge into a single value for orientation and position. So this is called orientation tuning. And this is super important. If you guys remember, this diagram on the right is actually an experiment where they give them different orientations of that black bar. And so you see from the top A and G, if you have like a vertical or horizontal, you don't get a lot of activity, so you don't get a lot of spikes. But as you're going and you're getting like super diagonal with letter D, you see a lot of activity. So this is this idea that you start getting a lot of like orientation tuning, and you, again, goes with the theme of like edge detection. You're like detecting something that's like out of the ordinary or like different. Okay. Did I skip one? Okay, and then so moving from the V1, you move to the V4, which again is a really important part that he talks about. So he talks about how the V4 compresses information about larger image regions with explicit signals for curvature. So he gets into a lot of the different like diagrams like this, which I'll talk about in a second, but that's kind of what I'm referring to right now. I'm gonna let you guys read this on your own, but basically like what's important is that V4 starts to detect these like areas of curvature. So again, like edges, we move from edges to orientation, and now we're doing curvature. So as you move through the path, you get increased receptive fields, which is why you're getting more information. And that was like when in lecture, you know how he talked about like the angles, like the degrees, like the degrees of, and then somebody like asked a question in lecture. I don't know if that rings any bells, but this is kind of talking about that like receptive field getting larger. And so he like throws this picture in a couple times, and it's just kind of to show like the scope of V1 versus V4. And then we take a brief intermission of from V4 just to talk about configural coding, because this is where it starts to like come into play. So basically, configural coding is when individual neurons represent object parts and their object relative positions. So ensembles of such neurons actually can represent entire objects. And he shows this diagram as like a sample of just this concept of configural coding. So configural coding is sort of a theory, and we'll talk about another theory later that he says and explains relative to like faces. So those are like the two big theories you should know. This is exactly from his slide. Wanted to include it because I think it's really important. Configural coding is a good concept because it does all these different things. It compresses, it makes it explicit, it makes it stable, and then it also does one more thing that he didn't list at the beginning of his lecture, and that is that it's really productive. So basically, if you have these neurons representing all these different parts, there's like a million different combinations of things you could come up with from that, which is why it's like really productive. Okay, so going back to V4, configural coding in V4, and going back to how V4 is a compression strategy, he talks about this specific example where you can see like in response to like curvature, you have like response of the V4 neurons, and that's kind of just detailing again this aspect of them representing curvature in particular locations. Okay, so these are all the experiments that he showed in relative or for V4, and basically from the start of his lecture he talks a lot about like 2D images, but to make sure that this is explicit representation of 3D, because that's what's like what you're viewing like in the real world, he moves to showing these 3D images that the V4 neurons actually respond to. And so the red border actually shows like spikes per second, like the response of the neuron. And you can see that for both lineages, you evolve like a high response shape, which has a concave curvature facing to the right. So if you look at the ones on the bottom left corner, you can see that kind of like concave curvature. So these particular V4 neurons that they're testing, like that's their tuning. And then he moves to talk about a couple other experiments, which I think I'm just going to leave up here for the sake of time. But if you have specific questions, like, feel free to ask afterwards. And then he does this thing with deep convolution network models. And so he talks about AlexNet. Super, super brief. This is like a very dumbed down definition. DCNs are multi-layer networks where multiple lower layer units feed into a single upper layer unit. And there's like nonlinearities with this. DCNs like AlexNet have been trained for recognition of object categories. And the important aspect of this is that he ties it back to his neural processing because these DCNs can be used as models for neural processing in stages of visual cortex. So I thought this was actually really, really cool. In his slides, he shows comparisons of like AlexNet's responses and a V4 neuron's responses, and they're actually pretty similar. So moving forward, he again shows this dog picture. And so what he's doing is again showing like the scope of the receptive field increasing and like what is being encompassed. So moving from V4, the next aspect is the TE, which has a large receptive field and therefore encompasses entire objects or multiple objects. And so the TE actually, again, this is very important, is an aspect that compresses information by encoding medial axis shape. And so it takes advantage of how real world objects have like strong regularity and symmetry across different axes. And so when you look at like the topmost, you see there's like a lot of red. That means it's all responding to a medial axis. But then if you move to some other TE neurons, they don't actually respond to only the medial axis. They're also sensitive to the surface shape of whatever's around the medial axis. Yes?
What is TE?
So I tried looking for what it stands for, and I literally think it's just called TE. So it's like after the V4 in the pathway. I'll see if I can find a better answer, but sorry about that. Okay, so moving forward, yes?
Yeah, so like by the time it gets to the TE, that's coding for an entire object, so that's like dog and dog-related shapes.
Yes, so it's like not belly of dog, but dog. Yeah, so I don't know. He includes that image. It kind of gives you an idea of like how the receptive field is like widening. But yeah, that's one way to say it. Yeah. Okay, so you know how I said with configural coding that was like one theory that like relates to these images and stuff. There's another one that's called similarity space coding, and so each neuron actually represents a continuum of similar shapes. And so similarity coding can be based off exemplars, which are defined as previously learned stimuli. And neurons encode similarity of current stimulus to their, it's current stimulus to their exemplar. So the sample that he gives is this map on the right. And so the new stimulus is considered the giraffe. And with this similarity space coding theory, your other animals are like your exemplars, or what it's like being compared to as like similar or different. And then so basically that's what it does. Like instead of having configural coding where like each neuron represents a certain aspect, now you have something that's like a continuum that you're able to compare as like similar shapes. And so basically this example is not actually super real. This is just a way to explain like similarity space coding. It hasn't been like proven with animals. But what it has been proven with is faces. So he talks about face recognition at the end of his second lecture. And so basically faces are encoded in similarity space centered on geometric averages of all faces that you've like seen in your life before. And faces are special object class because they have a high degree of shape similarity, making them really difficult to actually distinguish with configural coding, which is why this theory comes into play. And so he talks about, I think, this one lady's lab who does like really, really cool work. But I boiled it down to like this one experiment where I think shows what you guys need to know. The faces were basically parameterized, and then they used PCA. And what they did was they showed a lot of different spatial position of features to composite descriptors, and those represented like shape and appearance. And so with those, they were tested, like neurons were tested within this space. And STA is defined as the spike triggered average position. And so I know that was a lot, but basically that explains like how they got their x-axis. And then the firing rate is as you move along that continuum or within that space, you see the response of the neurons. Oops. Okay, so I'm going to briefly explain the graph on the right, but I think what's most important is that from that you can understand that there's, like with facial recognition, it's like not exemplar based, but rather ramp tuning in face similarity space. So you know how I talked about with the animals one, it was like exemplars with like the hippo and the whatever other animal it was. This is a little bit different with faces. So what you can tell is you see how there's like change across the x-axis, but there's no like change across the y-axis. That's how they show that it's not exemplar based, but rather ramp tuning. Oops. Yeah. Yeah, of course. Okay, so basically what they did was they looked at like a bunch of faces and they put parameters around them so that they could actually like boil it down to composite descriptors of very, I don't know how to explain it without being super confusing. So what they did was they found composite descriptors of how faces like vary in like shape and appearance in general, and they then tested neurons like within that space. So I guess a good way to actually describe it better is just to look at the image of how they have like the different faces, and how they get those different faces was like based on those composite descriptors where they like manipulate certain things. Does that make more sense? Okay. Okay, so what is cool about this is, or like I guess what's important about this is that similarity space coding actually compresses a large number of stimulus parameters into a tractable dimensionality. So this is like what's important from the facial recognition aspect of it, which is why it's actually really hard to describe structural differences of like different, like of familiar faces. And he talks about like describing like a teacup or a teapot versus like his grandmother. Like this is where that analogy plays in. And I just included this because I thought it was pretty cool. You can actually reconstruct a very close approximation of the original space, face, based on the pattern of responses of the neurons. And I thought that was just like super cool because they actually do look really similar, which is kind of scary. But these were not covered in lecture, but I did want to throw them up there just in case you thought it would give you more context to your studying. So what's important is that through this lecture, when I talked about like V1, V4 and everything, this was all the ventral pathway, which is what he calls the what pathway. And then your dorsal pathway is your Y pathway, but he doesn't cover that at all. Like you won't be tested on the dorsal, but just for context, make sure you understand that like what we covered was like the ventral. And then I think when you're done reviewing all of Connor's stuff and you go back and you look at all these different buzzwords, just understand like what fits in with what, and I think you'll have a really good grasp of his material. I've also gone through all of his exam problems, so if you have specific questions about those, like not the ones on your current exam, sorry, I like did not look at those, but the ones that are posted on Blackboard, I can answer questions from those.